Citizens honor. Thereby, the few minutes back, Dr. Swarnandhra is he is an expert in the cancer, the medical oncology, and he himself has given many ideas to the students. The area of the image processing, the area of chemical engineering, and many other areas that have been delivered with ideas. Definitely, I'm sure that the students can get the benefit of the knowledge. And today, the first session theme, as you know very well, just few minutes back, and Superstar has visited, from circular bioeconomy to cancer therapeutics, innovations that matter. That is the theme of the first session. In this session, we have three experts. Dr. C.H. Mohan Rao garu is a distinguished professor and head Department of Biological Sciences, BITS Pilani, Hyderabad campus, and also former director CCMB. And his lecture will be on why should engineers know biology. As you know well, even in the AICTE, they have mentioned that biology should be one of the subjects to be introduced in the first semester itself. But unfortunately, the universities are not following that. The biology is one of the very important subjects to be introduced even in the first semester of the BTech program. And I hope the coming the 2026 regulations, we hope the present Vice Chancellor will think about introducing the biology. And the second lecture is by Dr. S. Venkata Mohan and the director CSIR, Navy Nagpur, on industry ecology to sector bioeconomy, transformative solutions for sustainable development. And this lecture is in memory of Sri Dr. Elavati Nayadappa garu. And the third lecture is on challenges in design, discovery, and development of new cancer therapies. And the lecture is by Dr. Ahmad Kamal sir. He is a former outstanding scientist. I speak Hyderabad. The lecture is on challenges in design, discovery, and development of new cancer therapies. I'm happy to associate with this event. And I would like to introduce the first speaker, Dr. Sishek Mohan Rao. And
second is first.
second is first. Okay. I would like to introduce the second speaker, that is Dr. S. Venkata Mohan garu. As such, the biodata is much. Rather, I would like to try to break down the information. He holds the BTech in civil engineering and MTech in environmental engineering and a doctorate from Sri Venkateswara University, Tirupati. His academic and research credentials are further distinguished by international engagements, including a visiting professor at Kyoto University, Japan, and Alexander von Humboldt Fellow at Technical University of Munich, Germany. Dr. Mohan's research is positioned at the interface of environmental engineering, biotechnology, and sustainability science. His work focuses on developing regenerative, circular, and low carbon technologies. He has made pioneering contributions to acidogenesis, bioelectrogenesis, waste valorization, carbon neutral biorefineries, biohydrogen production, and CO2 bioconversion. His research has also significantly advanced an MES-based resource recovery, biopolymer production, and sustainable environmental biotechnologies. And Dr. Mohan has been the recipient of numerous honors, including the coveted last year Vijnan Puraskar, Vijnan Sri and Shantaswarup Bhatnagar Prize by the Government of India. Dr. Shantaswarup Bhatnagar Prize, the INAE SCRB Center, Abdul Kalam Technology Innovation National Fellowship, the DBT, Department of Biotechnology, Tata Innovation Fellowship, the vast industrial research area, the Key International Fellowship, the Prosper Net Scopus and Research Award, and many awards to his credits. And he earned his BTech in civil engineering and MTech in environmental engineering and PhD in the Sri Venkateswara University as a technique. And Dr. Mohan is a distinguished speaker today. I heartfully invite him to be with us to address our interest causes.
Thank you, Chair, for a nice introduction. First, I would like to thank JNTUK as well as two of the organizations who are conducting this important event. And also giving me the opportunity to present some of our work which we are doing at ICT and UV in the name of distinct lecture in memory of Padma Shri Dr. Yanavati Naidam. It is my honor to speak in his name. So just to give a tribute to Dr. Naidamma Padma Shri. So he's a visionary scientist, and with reference to CSR, where we came from media or ICTs from Council for Scientific Industry Research. So he is Director General of CSR, as well as Director for CSR CNR. So his contributions, major contribution looks into leather technology, where he tried to make it green process, which is at present also very relevant in the context of sustainability. And also he tried to link science and industry and promote self-reliance, which is nowadays a job. So it's my honor to speak in the name lecture of legendary personality. So today my presentation, I would like to present for 30 minutes. So initially I will try to speak about LERI, because what LERI is doing, and then few of the projects what we are doing in Andhra Pradesh. Then I speak a little bit about resource and environment challenge, and try to go into industrial ecology, circular economy, which are the emerging areas which needs to be considered for sustainability aspects. So CSR Nation Environmental Engineering Research Institute, which is stationed in Nagpur, started in 1958. The basic scope of the institute is looking to all environmental aspects and works in the interdisciplinary domain on all areas, starting from water, wastewater, to climate change. So we have 5 regional centers. We address sustainable development goals. And when we look into the journey, it started in 1958. Initially, it started to address the public health. So at that time, institute named Central Public Health Engineering Research Institute. So for almost 15 to 20 years, it tried to work on water well disinfection, developing chlorine tablets, and try to address all public health issues. And then in 1974, because environment domain is changing and it is encompassing many of the areas. So as Environmental Protection Act also came, its name was changed to Nation Environmental Engineering Research Institute. Some of the pioneering technologies which he has developed. So far, it has about 7 to 2 technologies, which are highly relevant now also. Chlorine tablets, which was used at that time, was also relevant right now, using in many of the STPs because of its simplicity. So the first technology, legacy technology by Lady, is an Alabota technique, which was developed to remove fluoride from drinking water. It's a proven technology trying to use parts, sand, and some kind of alumina coagulant, and it was demonstrated and established in many of the villages, initially in Nalgonda, in other areas also. So that is one very successful technology. Still in medical books, students studies about Nalgonda technique when fluoride comes. So after that, it has done many of the things, extension of Nalgonda technique to electroplurination to reduce the contaminants. Like that, many has been done. I will explain in between whenever So few of the projects at present which we are doing in Andhra Pradesh, one of the important project is at Kolleru Wildlife Sanctuary. So you know that the Kolleru is one of the very important thing with reference to biodiversity. So we are operating two projects, one for NGT and AP Pollution Control Board, to look into the water streams which is coming into the Kolleru and try to identify the actual topography of Kolleru. So quantifying the wastewater characteristics and looking into hotspots which drain is having more impact. So based on that environmental impact assessment and carrying capacity was going to give to AP Pollution Control Board. In another project, we try to look into groundwater, surface water and soil water in 5 of the contours of Kolleru Lake. This will try to look into the impact of polluted water on ground and soil contamination. This is also we are doing for Andhra Pradesh Pollution Control Board. Another important project in Andhra Pradesh, we are doing for 2 cities, Ongole and Guntur, to understand pollution, air pollution, with reference to where it is coming, the source approximation. So once we identify particulate matter 2.5 or particulate matter 10 or air quality index, that is not enough. But what is creating this particulate matter 2.5 and what are the chemical compositions that will help us to design and management air pollution in a very significant way. So these studies we are at present doing for Ongole as well as Guntur. So one more important project, Polavaram project. We are working with this since 2021 to understand landfills which is coming from Polavaram, how it is impacting environment. And this year we are going to submit the final report. So near to about this is NGT case again. NGT has given us this project to do. So this is with reference to the groundwater quality as well as air water quality. So one more project near to Rajahmundry, Andhra Pradesh paper bill. This is with reference to discharge of treated effluents from paper mill industry, where to dispose it without affecting the water quality. So we are trying to identify point where exactly this can be discharged so that it will not have much of the environment. So other projects also we are doing. I'm not going to be, but I would like to mention it's from ICT and I was earlier working as scientist. We have the municipalities, about 8 municipal corporations in Andhra Pradesh. They helped us to study COVID in wastewater, which subsequently has given a lot of inputs. At present, ICMR and One Health wastewater surveillance for multi-pathogens has been taken up, where Navy is also part right present. So we have done it now, Visakhapatnam, Krishna, Guntur, Anandpur, Vijayawada, Kurnool, Rajahmundry, Narsaraopet, and whatever concentration of COVID we are getting at that time is matching with the coherent studies which is done by the government. So to start with the presentation, I would like to show this slide. This slide says, which is published in Nature somewhere in December 2020. First time in human history, the man-made biomass increased the living biomass. So this is not sustainable because living biomass is very much required to regenerate back whatever we are doing. So we are producing more biomass than the existing living biomass. So that will have a lot of negative consequences. So this has doubled significantly in last 20 years. So normally living biomass, the plants and its constituents will be 90%, which has a significant potential to regenerate, followed by bacteria and microorganisms as well as animals, where humans also will be there. Near to 1% of this living biomass, animal biomass will be there. So for comparison sake, when we look into the animals biomass, it's about 4 gigatons. But plastic 8 gigatons. So see the difference. So where can we generate our whatever we do in here? And similarly, plants about 900 gigatons. But building and infrastructure 1.5. So this is not sustainable. So we depend for our resources, whatever materials we are using, we depend on positives, either for energy or for
तो
material.
इसका दो
Whatever material we are using,
तो मैं फाइल दिया
going for petroleum or
उसका दो
natural
स्लाइड्स
gas.
फर्स्ट
So
बताएंगे
without these
उसके बाद
feedstock,
इसको
our
करें
society cannot sustain.
ठीक है
And also, society
Agriculture
will call it as illegal.
इसमें का जो मैं वो
So completely our example,
Challenges
taking
का
from mother
पुराना
Earth,
डेट
using it
अप्रैल
and throwing it even though it has to be.
एमजी बोल के
So
रहे
this,
एमजी
our finite resources are
वो फाइल
like whatever
तो वो
was
continue.
not, it is a finite. So we will not have much material. Already, whatever material, the fossil material is getting exhausted, you can see this graph. So if you are using in this way, future generations will not have resources for their daily need. So when we are using materials, as I told, like when we look into the figures, the world consumes near to 100 gigatons of material per year as per 2021, which increased 4 times since 1970. But the population only doubled during this time. So in last 5 years, we used 500 gigatons. So when we equal the recycling, this is a report published by one of the circular economy agency. So resource recovery is significantly reducing. And you see from 2018, which was 9.1%, but this last year it was 16.9%. So we are wasting the resources either into landfills or other places. So that is not a sustainable practice. As I told, it's a linear economy. It's a kind of cradle to grave approach where we are taking resources and try to use it. Once we use it, we are throwing it to the landfills or some other places, even though it has a functional value. So this is creating a lot of environmental pollution. Whatever pollution we say, either greenhouse gas emissions, environmental, all these are because we are not properly using the materials and we are throwing it as a waste, which is subsequently creating all the pollution. So here I would like to mention about the planetary boundaries. So this was developed by Stockholm Center, Resilience Center. So when we have to address the environment, how healthy it is, there is no exact figures. So this will say with 9 parameters, we are crossing that particular boundary or we are at the same side. So we have about 9 boundaries on which this planetary boundaries was developed. So climate change, biosphere integrating, biogenic growth, land system change, fresh water usage, ocean acidification, atmospheric ozone loading, stratosphere ozone depletion, novel entities. So all the 7 of the 9 boundaries cross the safe operating space. That means we lost a lot of biodiversity, which we cannot get back again. Like that, so every parameter is going towards a negative. So except atmospheric aerosol loading, stratospheric ozone. This shows how complex, how challenging our situation is. So when we compare from 2009, there is 7 boundaries crossed, but 2025, 9 boundaries. So we have to change. Change is inevitable. So changing means population is going to increase from 8 billion to 10 billion by 2050. India particularly have 18% of the global population. So we need more resources and global domestic material consumption was about 40 billion tons per year in 2010, which increased to 100 billion tons. When we look into the person, 5 to 27 tons per person per year, depends on the economy of the country. This is the global figure. So by 2050, we need 180 billion tons per year of resources to support our economy. So if we continue to do like this, we require 2 times more water, 2 times more fiber, as well as 2 times more food. And adding to all these things, nearly 70% by 2050 will be living in urban areas. So that is again a very major concentrated package where significant resources will be used, significant emissions for pollution will be coming. So 75% of natural resources will be used there, apart from 50% of global water product, waste will be produced from these places. So this transition is very important. I would like to say we have to go with 1.5 degrees centigrade and we have to go to net zero by 2070. We have to decarbonize our process and we have to reduce dependency on fossils, either for materials or for energy. And we have to infuse renewables as much as possible. And also we have to adopt industrial ecology, which is the concept which is emerging, and we'll discuss about that. And also we have to infuse nature-based solutions. Always when we integrate a nature-based solutions, it is a regenerative nature, which will get back what we have done. So you might be going yesterday. So we are crossing yesterday. Now in 2017 we did 1 year to regenerate that, but now it became 1.7 years at present to regenerate that. So nature-based solutions, the importance was given in 6th IPCC, International Panel on Climate Change, where they suggested to go with circular economy and also with nature-based solutions. It's a science-based process. So then we are adopting nature-based solutions. They have given some graph. If we are going as usual, we'll be reaching somewhere here with respect to the carbon emissions, but we adopt it in a very significant way. We can reduce our emissions. So nature-based solutions have significant impact. Here I would like to say industry ecology, which is a concept of which I want to present. This industry ecology is a interdisciplinary concept where it looks at the metabolism of any system, whether an industrial system or a urban city, to understand material flow in the system and what material, where material is stocking in the system and what is coming as a waste. So this will help us to understand material flow and understand where waste is going and try to close the loops, which is very important in order to reuse the resource. So here we can use many of the tools like life cycle analysis or material flow analysis and come up with eco design, which is very much required. So transition from linear to circular economy is one of the major things in industrial ecology. So the basic principle is on using the material and design the material in the form of a product or a waste which is coming out of that process is a feedstock for next process. So like that, we can circulate material as much as possible so that we can reduce waste from the system. This concept is very important to keep the material use as much as time possible with minimal waste. So here biological routes of conversion is very important because biological routes are able to produce the materials by using the sustainable routes. So these biological routes I am going to discuss today. So this is with respect to waste. When looking at circular economy, waste is very much important. Is very much important feedstock which needs to be addressed. India has huge amount of wastewater, industrial water, wastewater, domestic wastewater, domestic municipal solid waste, which are not tapped. Only 5% of resource recovery at present we are doing, but we have a significant potential to use. So when we look at the urban metabolism, that is cities, there are a lot of loops which are open in the form of technical, biological, and water and fuel, which is going as a waste, even though it has the potential resources. This also needs to be regulated. So how we can regulate when wastewater is there? Normally, when we go with chemical process, we try to remove the using some chemical, the important nutrients present in wastewater, and it will go as a hazardous waste. Instead of that, when we are going to biological food, then we can convert into some products like aerobic process. When we give oxygen to aerobic bacteria, it will convert into new cells. If such will come, we can use that cell. But aerobic process is energy giving process. Aerobic is energy intensive process. So here, conventionally, when we go for methane production, we can produce methane from organic nitrogenic bacteria, and then we'll get CO2 and hydrogenous entity. Otherwise, we can regulate nitrogenesis, we can go with acidogenesis, we can produce hydrogen along with volatile fatty acids, which is the building block chemicals. Or otherwise, we can go with the recent techniques which are coming bioelectrogenesis. We can produce organic carbon into energy in the form of bioelectricity apart from producing oxygen. Another important route, photosynthesis, where when we use the photosynthesis like microalgae, which will convert both inorganic as well as organic carbon, that is CO2 as well as organic carbon, into biomass, apart from oxygen treating the wastewater. So likewise, we can produce many of the chemicals or products or biofuels by anaerobic process. like converting the waste streams. When we have a solid waste, it's a different story. 50% of Indian solid waste is biodegradable in nature. So basically, what is doing, we are sending near to 90% to landfill. That is creating a lot of issues like methane generation and other things, greenhouse gas emissions. Little bit going for composting, very little, and a little more for incineration for energy recovery and digestion for biogas methane. But we have significant scope here to convert by little bit pre-treatment, we can convert into sugars and we can use for industrial chemicals. Or we can ferment to using some of the techniques which I have shown earlier, either to produce energy or to produce intermediate products which can be subsequently converted into chemicals. So likewise, carbon is again one of the important greenhouse gas. So basically, algae is responsible for much of the conversion of CO2 into oxygen along with biomass. But this is a slow process. When we want to go for industrial way, so it is not feasible. It's like 10 to 12 days it required to convert. But in the now, certain group of bacteria are there, so that we can source from anaerobic process. We can take that bacteria and we can convert that CO2, inorganic CO2, by legal pathway into value. So this slide I would like to show that whatever waste we take, either domestic wastewater or industrial wastewater or solid waste or CO2, we have many of the bioprocess at different stages. Anaerobic digestion is a mature process which at present using. Apart from there are different processes which are at different PR meters that can convert into bioproducts, different kinds of bioproducts, which have significant application apart from waste management. In this way, we can close the loop, and can use the material product into the system again, so that the emissions or pollution generated because of not using these materials can reduce. And apart from that, we can use these products, reducing our dependency on fossil. So what are the in ISPT we are going to is some hydrogen production. As you know, hydrogen now is one of the important energy vector, which is going to reduce when it is taken into the system significant amount of greenhouse gas. At present, it is again produced by fossil fuels, but in ICT we developed a process with estrogenesis to produce hydrogen by using municipal solid waste, that is biodegradable solid waste. So this we have established with the pilot plant 10 metric capacity with a biorefinery approach, where every product coming from the process will be used to produce other kinds of products, so that the total system can be closed by multiple products. So one of the circular economy thing, one of the major issue with respect to urban or rural context is fecal sludge. So now handling fecal sludge in with septic tanks where the decentralized treatment systems are not there. So that is creating a lot of problem. So we can use this fecal sludge, very good thermal process, very less energy intensive thermal process into organic material. So this will close the loop, like this organic material doesn't go for agriculture, or we can use it for the soil. So we'll be closing the loops without affecting. Otherwise, when send sludge to the environment, dispose wherever this sludge in the environment, it will create a lot of health and environmental impact. So one more closed loop operation. Normally, thermal power plants, they produce a lot of fly ash. So fly ash, handling of fly ash, part of it is going for cement manufacturing, but others are lying around the fly ash dumps, right? So Lily has developed a technology where these fly ash lands were contaminated sites where region will get back by eco-technology, eco-regeneration technology, where simple waste material is used as in the for plant production using some specific bamboo plants. So far we have to last 5 years we have 2,000 acres of contaminated land, either with fly ash or in other contaminant was regenerated. So it is again closing the circle. So not opening the loop. We are trying to close the loop by converting it into bamboo, and that bamboo has multiple applications apart from regenerating them. So these are the different climatic conditions where it has been used, this technology used. And inauguration of 92 species of bamboo recently on DG Medal has opened in the campus. We have clear to 93 bamboo species all around in Germany. So here our AP, you know that we have a chemical and pharma industry, one of the industrial sector. It is always challenging to address environmental issues of chemical-based industry. So as we are not going to be, but industry is the second largest greenhouse gas emitter. And when we look into the industry, chemical industry particularly is responsible for 50% of the 25% of the total emissions from industry, and among the 15% is from ammonia industry because it's in greenhouse. So the basic issue is it's an open loop system where we try to take resources. It's a resource intensive, energy intensive because of the emission system. So, so we are trying to make the material, and there is no chance of going with circularity. Production, then package, and the end of the pipe. Wastewater we do at the end of the pipe. But unused chemicals we are not able to address so far. So this needs to be addressed because it has lot of, every industry has carbon intensity, significant carbon intensity. Mostly it is with ammonia and hydrogen because of issue with the display, we are not able to see ammonia about 3 kg CO2 equivalents per kg product. But hydrogen production by grey beams is about 10 kg CO2 equivalent. So a lot of environmental challenges, particularly it contributes to 7% of total global greenhouse gas emissions. It depends 10% on global energy, 50% of feedstock. This is the global context. And used water, 10% of water is for the this purpose. So generate significant amount of wastewater, hazardous waste, again UC air pollution emissions. And these will create a lot of environmental issues as we know. And I will try to address fossil-based feedstock dependency, how we can address this. So for sustainability with respect to the chemical industry, to make it sustainable, we have to address raw material. We have to infuse renewable energy. We have to infuse renewable non-fossil based resources. And in process, we have to adapt green chemistry principles as well as eco design. And end of the pipe, we have to go with net zero emissions, not releasing anything into the environment, or repurposing or recycling or adopting biorefinery concept or industrial performance. So this is one strategy we propose in ICT, one of the projects using ISO 14044, how we can address sustainability in chemical process industry while adopting life cycle analysis as well as planetary boundaries, and circular economy is a major effect. So based on this, we are trying to propose a concept, circular interventions in chemistry. It's a concept again trying to infuse the circular economy. So the basic principle here again is to use these are the key factors where industrial synthesis is very important, which I would like to explain. It has to go with the synthesis or chemistry, where all chemical reactions will not be with the chemical based materials. Few of them we are adopting bio-based chemicals, then it has advantage. Have to go with industrial technology, biorefinery as well as biomanufacturing, trying to use biorefinery, biomanufacturing to produce chemicals and has to adapt to CCUS. So biomanufacturing as biology students, they know much hype, or I cannot say it's a hype. A lot of interest is there from DPT funding, lot of funding, because they are trying to use biological routes to produce chemicals, to produce fertilizers and many of the products. Even in the space research, they want to adapt biological routes. So we have advantage of different kinds of bacteria, the different kinds of microorganisms. here it is not clear, but you can see here that many of these reactions or many of the chemicals we are using in our chemical process can be produced biologically by these based on fermentation, organ based, or with molecular pathway converting CO2 or CO2 and even sweet oxidation. So this needs to be adopted as much as possible so that we can reduce the impact. This graph shows how we are producing a chemical by fossil as well as a chemical by biological goods, how significantly we can reduce greenhouse gas listed here from IEA. So again, the biomass is a very important feedstock here where we can use it for multiple purposes. So we can convert it to cellulose, we can convert it into liquid, we can convert it to heavy cellulose. And that will become platform products for many of the chemical reactions. So once we start doing it biomaterial, then it's more sustainability we can increase. That's what this biomass biorefined this is. And when plastics are there, at present we have huge amount of plastics. But what some of the people, like some of the companies are doing, trying to convert it to energy. So we are trying to the functionality of chemical, we are reducing its functionality, try to make it to energy form. So we are losing a lot of important feedstock. Now the concept is upcycling the plastics, either by chemical food or biological. Upcycling means try to convert the basic plastic material into some intermediate chemical, either that can be used for again plastic production or in another process. So that we are giving value to the plastics. So we are trying to directly convert to energy. We will not get any chemical. We are using a good amount of fossil material. Again, we have to depend on fossils. So likewise, CCU, CO2, I explained earlier, which we are doing in ISCT. We have a pilot plant. Nuclear pathway we are trying to use and try to convert CO2 without giving hydrogen. Hydrogen is required to convert to fatty acids, but we are trying to infuse electrons and trying to convert into different products. One of the important products is biosuccinic acid. This is the only biological group, bacterial group that can take CO, that requires CO2 to convert to succinic acid. So this thing we are trying to develop with our isolated strain. It's a reverse PCA pathway where when CO2 is given, then this reaction will happen. Biosuccinic acid we can produce. And this, when we compare with the chemical group, at present they are producing with chemicals, the intensity of CO2 emission can be significantly reduced. And here you can see climate change is going in a positive way, negative side. But chemicals, we are having a lot of impact. Because we are using CO2 here, so there is no climate change. And on planetary boundaries also, biological group has all positive things, but the chemical group has some negative things. Likewise, we can produce many of the chemicals which needs to be taken up by young scientists like you, because future is requiring bio-based materials which are sustainable, and developing innovations in sustainable production or sustainable practices is very much important. And here this is the model where iodifying the model where waste from one group can be used for other products. Like for example, this is the last slide I would like to explain. When we take wastewater or water, then we take if we directly go to one of the initial reactor, pre-treatment reactors, then go to acidogenic, where we can produce hydrogen. The outlet will contain fatty acids that can go to any of the process, like photohydrogen or hydrogenases for methane, so that complete carbon will be converted, or bioplastics production, or electricity production, or algal cultivation. BFA is a good feedstock for algal. So with algal cultivation, we get CO2, and the biomass can be converted into biochar. So by 3 stages, 90% of carbon will be converted into some product, and then that will go for a closed loop after going to the plantation. This biochar will also go for plant. So we can close the loop. So by this way, we can establish a sustainable loop. That's what I would like to say. This is the biorefinery which is available in pilot as a pilot plant in ISCT, but now not operational. We want to take up to next step. So this is my last slide. I would like to say Industrial ecology address circular economy, which is an important component, apart from industrial symbiosis. So industrial symbiosis, the classic example is Karlberg industrial symbiosis park in Denmark. That's the first and foremost successful example to show industrial symbiosis. Industrial symbiosis means that a cluster of industries will be formed in an industrial area where one product from one process will be used as a feedstock in other process. Likewise, in this industry sector, there is a pharma industry, the thermal power generation industry, aqua farming, as well as fertilizer industry. All these are integrated. The heat energy waste will go for pharma industry. The nitrogen wastewater will go for aquaculture. Likewise, everything is a closed loop, trying to make a synergy between industry to industry to reduce emissions. So this is one of the classic examples. When we look into either urban area or industrial area, if you can design in that way, then we can increase the sustainability of process as well as we can increase the overall we can reduce the environmental emissions significantly. This is the concept we try to make for urban biological materials, closed loop concept to close the open loops, where all the open loops are tried to close by green loops, which was here. So this was published, but these concepts, if you can take, then we can have sustainability. That's what I would like to do. So all chemicals which I have shown the first graph, you can produce by these loops, what we have explained. So everything we can produce, but this needs to be developed. But there are a lot of challenges which needs to be addressed. That's the young generation. You can look into the future. You don't have resources, but yours is a resource dependent society. Sustainability will be always there. So to address the sustainability, we have to come up with some kind of innovation, either biology, either engineering, or any of the domain. We can address these aspects significantly. So a lot of things has to be done. And future is, I would like to say, industrial economy has to play a major role for sustainability. I would like to thank my students whose work presented and funding from different funding agencies. Thank you very much for the kind attention.
Thank you very much, sir. I'm sure that the students and the scholars definitely going to come. And any questions, I request you to write the questions to the expert. You can as well interact with the data also. Anyone, please, please. From the thermal power plant, ready process are almost 3 hours.
Huge amount of fly ash generated because coal once it comes to fly ash, contain good amount of silicon and other heavy metals. So the process, it's a simple process where you can use some plant species who use of bamboo, but you can use the native species of bamboo to convert that fly ash into that silica will be used for growth of bamboo. That's why bamboo is important. And you have to keep microorganisms at root because root is very important initially for growth. We also have to keep organic waste either from distillery industry or any other things. And we have to give water for a certain period of time, 2 to 3 months. So once plants start growing, it will within 2 to 3 years of time, the contaminated wastage is taking so wastage
అది టెస్ట్ కూర్చుంటుంది
treated waste is coming from daily paper. So again, the disposal point is somewhere. So the pollution control board want to study alternative disposal point based on some of the factors. So it will not affect the environment or other environments. With that angle, they're trying to look into 2-3 other points where treated water can be discharged to the Godavari. Sir, is there any use of that disconfirm material? The use of that disconfirm material in an alternate of that. Which material? Disconfirm material. Yes. Dispensing material. Oh, paper is that? Yes, sir. Paper means now lot of because came to convert black liquor also. So now paper industry.
उधर तो नहीं खुलता ना
Yeah.
सिर्फ यहाँ के
So like a subsidy. So again, going for blue feedstock, we have a lot of
ये करें ना
issues. Instead of that,
ये पहले अपने ये इसका सिर्फ 2 स्लाइड आएगा अब पहले रिपोर्ट करें तो इसका कंप्लीट आएगा ठीक है वो 2 क्या 2 बंद कर देंगे इसका 2
Thank you, sir. And your visits today, useful to our students as well as faculty.
प्राइस बताया
Yes, sir.
Sir, today we have another expert, and he is Dr. C.H. Mohan Rao. Huh? Dr. Ahmad Kamal. And the morning only, I'm happy to get introduced to him. And he is one eminent professor. And I would like to introduce him briefly. And his lecture is on in memory of Padma Bhushan, Dr. A. Angelity. I think many of us know. He's the founder of Dr. Reddy's Lab. Dr. Ahmad Kamal is the NRF Prime Minister Chair and former outstanding scientist, sitting in Hyderabad. His lecture is on challenges in the design, discovery, and development of new cancer therapeutics. And most of the lectures relate to the cancer because that is one problem which presently people are facing. And definitely, this lecture will be very useful. And I would like to briefly introduce Dr. Ahmad Kamal. Dr. Ahmad Kamal is a distinguished Indian scientist and academician known for his major contributions to medical chemistry and anti-cancer drug research. And he currently serves as a Prime Minister Professor of the Anusandhan National Research Foundation, the Department of Chemistry, Osmania University. Born in Hyderabad, he earned his degrees in chemistry from Osmania University and completed his PhD in medicinal chemistry from Aligarh Muslim University. He spent over 3 decades at CSIR, Indian Institute of Chemical Technology, where his research focused on gene targeting compounds and nature-inspired anti-cancer agents. And he later served as director of National Institute of Pharmaceutical Education and Research, Hyderabad, and pro-vice chancellor of Jamia Hamdard. Professor Kamal has published more than 580 research papers, also 430 patents. I heartfully congratulate you from JNTUK. And it has guided more than 115 PhD scholars. Excellent, sir. We are happy for your presence. Several of his patent discoveries have been licensed for clinical development. He has received numerous prestigious awards, including the CSIR Young Scientist Award and the Randback Steel Research Award. He is also a fellow of the Royal Society of Chemistry and National Academy of Science, India. Sir, we are proud of you and really Your presence has elevated the glory of JNTUK KAKINADA. Heartfully welcome you, sir. And I think you will present about that.
Very good afternoon. I will be speaking about, as mentioned by the chair, on the how IFRAC is discovered. So I'll go with more of a layman's view rather than getting into too much of technicalities. But I will also touch upon a little bit because I must tell you that I took an impression because JNTU has a very strong pharmaceutical chemistry, pharmacy group and also biotechnology groups. So maybe I'm not sure whether such things are there in this JNTU because I was associated with JNTU Hyderabad for almost 8-9 years as an executive council member there. And so that's how I had that impression. But anyway, I will try to be as brief as possible. And moreover, I would say that I'm extremely honored to give a talk in memory of the Conclave. He has been a role model for a large number of, I think, from the pharmaceutical sciences side. This particular picture, what you are seeing, is his autobiography, which was released, in fact, after his demise. So both his son and son-in-law had released this. And a lot of things are mentioned in this autobiography. I will just touch upon 2-3 important aspects. How the whole process actually happened. See, he was basically chemical engineer from his educational point of view and was working initially at the Indian Drugs Pharmaceuticals Limited, which is very commonly known as IDP of Hyderabad at Swarnandhra. And within, I think, a couple of years, he mastered many things because he was working on mostly in process chemistry. And probably they got an idea that they have to start something of their own. So in a modest way, he started with a couple of small companies. One of them happens to be Standard Organics and so on and so forth. And then based on that, I think he got an art. How to really do the business with respect to bulk drugs. Because IDPL, as you all know, at one time had been one of the Asia's largest, particularly the sulfur drugs manufacturing unit. And it started becoming sick. So that was the time I think these people decided to start on their own. And that's how it got evolved into Dr. Reddy's laboratories. And this is where I think he has really made a mark. So as a businessman, definitely he has made a mark. But there was one thing, he was highly passionate about new drug discovery. Because what he was doing, or rather they were doing in this whole company, Virati Labs, was making generic drugs for the Indian markets, as well as exporting to many countries. And then were making lots of profits based on the good technologies they had developed for their processes. And that was the also time when practically we were not respecting the international patent law, because it was, I think, during the, this is Indira Gandhi's time when she was the Prime Minister, some of the pharmaceutical lobby had convinced her that not to have international, honor the international patent law. Otherwise, the Indians have to pay big money for purchasing any drugs. And that's really worked.
Although in 2000 and all that, it got changed. So all this, and after that, many other companies started cropping up in Hyderabad and around for making bulk drugs. So today we have so many, I mean, Aurobindo to Hetero, so many of these. And that's how, but that way, Dr. Anjali, I must say, he was so passionate about new drug discovery. Somehow, with a lot of this, although his family members were not very much in this thing, because as you know, new drug discovery is highly risky process. Because something may be discovered to some level, but at various levels it has to go through. So that's why some of his family members were not very much in favor of. But in spite of that, once he started in Hyderabad as the DRF, commonly known as, I think quite a big group was made, and the work was happening in two aspects. One was in the blood sugar reducing agents, and the other was in cancer. So these two areas they worked, and they came up to very good level, and a lot of international patents were filed on many of these aspects. And they wanted to move up to, I think, they finished the preclinical and all that. They wanted to get into phase 1, phase 2, and so on. But during that time, I think not this showed interest in the patents what he has filed for blood sugar reducing patents, and that's how somehow they licensed this to the Novo this with some good, reasonably good money. And these things were happening, but although I would say eventually it could not continue for very long. After some time, this particular thing got closed, and rather the one which is in the Bangalore as origin, they were also doing to some extent, but later that they all started working on definitely the discovery aspects, but taking a contract job since they have the expertise and all that. So talking about him now, I would say that he was somebody who could be a great inspiration, as I mentioned, in the pharma and drug industry. So today I will just cover something on how the drug is discovered very deeply, as I mentioned, because see, when you want to discover a drug, you have to understand that particularly depends on again the disease you are looking for. So you need to understand where the drug is going to act, and for that you need the right biological target. So these are different steps. Okay. So before going to this, I'll just give me a brief. So, how really, I mean, the medicinal chemistry got evolved. We can see that, the humans were practically a sort of culturally, people were trying to grow. Even in the oldest times, they wanted to understand how can we treat a particular problem, which we call it as a disease. So they started using various methods. And that's where I would say that was what we call as the ancient period. Although it also belongs to that area at the time. And Egyptian medicines, Babylonian medicines, and Chinese medicines. These were the things which were very much practiced at that time. And in the medieval period, again, we got Unani medicines and medieval medicines. the difference between this aspect and this is, say for instance, in Ayurveda, everything was being used, including natural products to certain minerals and metals and so on and so forth. But this particular thing, Unani, as the name indicates, this is an area which was, the expertise was developed in Greece. So there Unani means, Yunan is basically nothing but Greece in Persian. So that is how the name Unani has come. And this travels from Greece towards the African countries and the Middle Eastern countries and even up to the Indian one. So this is how these things develop. But the most interesting period is between 19th and 21st century, where it was the real evolution of, I would say, the allopathic drugs started. And how people started, because many taken also from folklore, what certain tribes were using, how the things were happening. By eating certain natural product, as you know, one of the big, great discoveries of aspirin. It's from the willow plant bark. That's how salicylic acid was there, and that's how that was being used for different things. So people started modifying those things. And lot of natural products and a lot of natural products, not only products, but natural product chemistry also being seen all over the country. I mean, all over the world, and even in India. So that is how this revolution happened. And in 21st century, the main thing happened is one started to understand how the drug mechanism happens and how do you then based on that one should rationally design the drug. And that is where we have so many things. I have not narrated everything in this. Fractional drug design, what we call it as nanomedicines, advanced. You see, nanomedicine today it's become very popular with respect to the nano delivery aspects, including the slow release. The many drugs which you use also are available in slow release aspects. KAKINADA is one of the pioneers in India on this. So these are some things which happen. But much more than this today, I would say, people are not only using just the computer modeling aspects, but many companies, many institutes have also been using as AI is being used in many aspects, even in drug discovery. And all these techniques are also being used. But how far we will have success, we have to see. Now, as I say, this is the aspect of targeted therapies. That means you try to target very specifically so that you don't disturb the other bio environment when the drug is being taken. Now, I'll give you some example here of this particular.
Next slide. Some. Okay, sir. Okay.
Sorry, I think
Sir.
एक मिनट पीछे खोलो
Yes, sir.
और नीचे जाओ बस
This is
एक यहाँ से रखो बस ओके
PPT got mixed up. See, as I was mentioning about the drug discovery aspects, mostly one part is the discovery, the other part is the development. And discovery comprises of the target identification, lead optimization, preclinical study. Whereas the development is mostly clinical, with phase 1 to phase 4 clinical trials. Now, with respect to target identification, one does with many techniques. And today, as you know, the aspects like genomics, proteomics, are playing in this. And we know the great advances which have been made in the area of molecular biology and so on. Now, lead identification is you're taking certain chemical compounds, either through synthesis or some modification of an actual product, we call it as a synthesis. And you're trying to observe how basically it's being basically it's bound to the target. So many of the natural product compounds, which are today what you say, particularly in anti-cancer, I would say large number of compounds are based on natural products. You talk of Taxol, you talk of Campusil, you can talk of many drugs like etoposide, renoposide, several of them, both based on again molecular toxin type of systems. They are again natural products like lignins. So you try to understand that what type of mechanism they work. So that is how it happens. That once a library is built, you shortlist them based on the activities, and then you basically take the ones which are the most active ones, and then you try to evaluate it further. So these are the different steps you go through, and you need to again make larger libraries of these compounds, because many a times you notice that certain groupings, certain things, how the interactions happen in that particular target. And that's how many people have used a lot of high throughput screening in between, the chemical libraries are made automatic. I mean, with basically robotic and thousands of molecules were generated. Although there were issues in that also, but what was thought was that you are consuming lesser time to make that many manually. So all these aspects were tried. And this is what happens during the rigor, right? And you pick up the best ones. And then you try to optimize it, which basically shows the best activity. And many, to understand again, it's interaction with the target. So you have to use many other aspects, because again, I must tell you that drug discovery again is not one specific science. It is chemical sciences, biological sciences, genetics, microbiology. I mean, so many things are included in it. And you need a really team who are specialists in all of them. And finally, you also understand that how this particular molecule, when it is targeting with respect to the specific protein or an enzyme, then based on that, apart from the effect, I mean, there are two aspects. One is the biological effect, what you are getting. So for instance, I'll tell you an example. Something you're looking for, say, blood pressure reducing molecule. So you physically are seeing in a model, animal model, how the blood pressure is getting. Is it or reducing or not? Then the second thing what you will be seeing is what type of mechanism, which are the actual targets. So for instance, there are a large number of blood pressure reducing agents based on beta-adrenergic protein, which are called as. So actually target is affecting. So like this, there are several examples for different diseases and so on. Then you basically now you have to do studies not only in cell culture but also in animal models. And after that, you basically pick up the actual promising compound or the most potential compound, which is showing the best result. And for that, we will do the ADME studies again, the absorption, distribution, metabolism, all these explanation, all these studies are done. So these are part of again heat optimization aspects. Now this is once you succeed all this, you go into the preclinical studies. So in the preclinical studies, you basically see the therapeutic effectiveness of this ability to be, which is not yet administered, it to be administered. So this you try to see within the animals. So that's how we call the ones which are there. This is the in vivo, and these properties not only the actual pharmacological effects, but its toxicology and other things are also studied, because you also need to know that what type of other effects will come based on these. So these are some things make one will do. I'm sure this will be good for the students. I know many of you know all this. Then we have, once we identify, we quantify the molecule and also understand its metabolites. We take this and try to develop the dosages, formulations and other things, how to make it better. The other one important thing is, many drugs, when you come up to this level also, you may get great success with respect to the biological activity and so on. But they fail when you are trying to give it orally to an animal. So that is where the bioavailability is assessed also. And if it's not going through very well, you try to use certain formulations to make it, or to modify the structure, covering in certain groups which may not alter the activity, but it can enhance the bioavailability. So these are some things one would do. Then basically, after all this, 3 to 4 or maybe 2 to 3 molecules are shortlisted as to be taken for potential the actual phase studies in the clinical. Why you need little more is because many a times, even though it's everything is successful till this stage, but once it gets into clinic, all may not become successful. Some may fail at phase 1, some may fail at phase 2, depending. So then you keep always some standby. So this is where it happens. Then, see in phase 1, clinical studies, it's basically you are seeing the molecules only for their basically toxicity. You're not, and that is how you are using the volunteers who are not really patients. They should be healthy volunteers, just to understand that what this molecule could attack for. So that is where I think once it succeeds this, we can go to the next stages. And also you try to see many other things, like how they could differ with respect to male, female, because again there could be hormonal differences. So there could, one has to understand that. Even not only that, I mean, it will have even the racial differences also. I mean, certain drugs have been successful on certain races, but when you look into certain races, it created some problems and it cannot be successful. So these are some things one would go. So once you succeed in this, you go into the phase 2 clinical trials. And here, phase 2 clinical trials will have volunteers. I mean, this will be the actual patients, with a specific disease. So you will have at least 100 to 500 patients in each group when you are going to do this study. And here you analyze the short-term toxicity effects, and then once you know that they have an optimal dosage, doing very well, the effect you are looking for in these conditions, you take that when it is frequent to determine and so on. Now, in case preclinical studies, this is, I would say, a very interesting study. What you do in this is, here, one uses volunteers who are patients and also who are practically the patients which which will not be administered the actual this molecule which is being examined. You give a molecule, they call it as a drug which is called as placebo, which has nothing. But that placebo also works because many people have a psychological effect with respect to this. So that is where this study is also very important, because in this case study, neither the patient knows what he is getting, either it is a drug or a placebo, nor the doctor knows what he is getting, because these are quoted practically. Mainly because to see that there is no bias in this. So that's how the molecule passes through. It will just rush through certain things. The phase 3 with this study, and finally we get into phase 4 clinical trials and everything goes well. Here you do a little more, number of patients will be much more, formulations will be found, new type of formulations will also be examined. Then you also try to examine the processes what you are making this molecule to the chemistry, whether it is viable, to what level, how far you can do the large scale, and what could be the cost. And the most important thing is, when any multinational or most of the companies who have been doing new molecules, they have to understand the cost aspect of the final. So when a new drug is put in the market, it will not be the actual cost of the raw materials or this, that is not the cost. The cost will include what all research they have done up to the phase 4 clinical trials. So you will see the cost, how they spend, approximate cost. So based on that, they will try to fix the cost of the drug. So that is why, because this, otherwise they can't do research tomorrow. After finishing this, there is no possibility. I mean, so that is how many times we have been seeing that in the US and most of the European countries, most of the drugs of that level are highly expensive compared to our Indian standards. So this is what you can see the pipeline here. So from practically the research phase, what you start for the discovery, for identifying actual, we call it as new chemical entity, NCE. It should take about 1 to 3 years. And for preclinical development will also be, this will be IND package, what we call, and this will be also another 1 to 3 years. And the clinical development will take about 3 to 6 years. So approximately, yes, till the new drug application is filed, whether to an control India or FDA, you have spent, I would say, something like 9 to 14 years. And this is where I think discovering a drug, it just cannot happen in a shorter time than this. Although there are little shortcuts for particularly areas like cancer and so on, to some extent, but still the long-term effects and other things to cover up, it takes the speed. And you can maybe, then finally it goes to marketing. But you can see in this, I mean, it would be something like the whole process could take quite a large money. So that is where the whole process is remarkably expensive, I would say. I will skip this. I'll just touch upon, I think, couple of I mean, it's more like a story of our work, what we have done with respect to the discovery of new cancer therapeutics. And I'm just giving you one specific aspect of this story. Although we had worked in, I would say, more than 6 to 7 target areas. This is a specific area where we have been working on this for just DNA minor group binding agents. And on slide, what you see is basically these are the structures. These are what we generally call as apomycins or pyrrolobenzodiazepines. These are the tricyclic compounds, which are basically isolated from the Streptomyces species. And Anthramycin has gone up to phase 2 clinical trials and it failed because of cardiotoxicity. And same is the case with respect to tobramycin, which has also completed phase 1 trials but could not proceed because of its high side effects. So this was this was something which we thought is something where we could do a total synthesis of these molecules. And that will give us a handle to modify them accordingly. Because we knew, so for doing that, there are two aspects. One is you should have a good mechanism to synthesize. That means you should explore the methods of synthesis, how to make such a molecule. That is one thing. Then to make a library in such a way that what type of functionalities you want to change. So, but before that, also you need to understand the structure and relationship. So I'll just go through some very quickly. So this is the molecule, what I said. You are all aware of major group and minor group of DNA. And this is the molecule which gets into the minor group of DNA. And once it binds there, it basically that is the where once it's covalently bound, it starts, that's the kill mechanism which starts. Now there is another interesting aspect in this series of compounds. While it's binding, there is a base pair sequence selectivity in the DNA. And this is where many research groups were looking into, I would say almost 2 to 3 other research groups are working in the US and UK. And we were also there to get certain molecules which have a preferential sequence selectivity, and selectivity should be for as many number of base pairs as possible. The understanding was the more it will have sequence selectivity, then it could differ from something where this molecule, the difference in the normal cell and the cancer cell, although there is not a very clear-cut thing that time, and one you're having much more of this sequence selectivity. It was understood that it could have better much more selective to the cancer cells. And that's how this search started. And as I mentioned, this binds into the minor group, where the NH2 of the Golgi is basically forms a covalent bond with this heme functionality. And that's how this attack is formed. So once you have this, you have to understand the structure activity relationship. As I mentioned, and also this is just a representation of and what you say here is the anthramycin which is shown in yellow, that how nicely basically fits inside the minor group of DNA. Now, this is, I won't go into all these details. We had to work on the strategy for synthesizing this. So several methodologies were developed. I won't go into all these details. Let me be very quick on this. These are a couple of different strategies were done from a literature point of view. When we started a new approach, which is different and much more easy emphasized, so that we need to make large number of molecules to such these type of methodologies. So various methodologies were developed by us, including the comment-based, but that was very popular in big libraries. And also like instances where we have used resin-based reagents. Again, this is from the green chemistry point of view and eco-friendly type of process, where you can always reuse these reagents after recovering them. So these are, now I just talk about very briefly about this. See, you have seen that already in that picture, that this particular imine bond, what we call it as, is highly essential because this is the one which forms, which allows it to form the NH2 of the guanine equivalent. So this is where it happens. So these are a couple of things which we have to keep in mind, including the H stereochemistry in this class of compounds. Why this H is sitting on the top? It's the S stereochemistry. That means when you have this structure, this scientifically, you can understand this like a half open lid of a envelope, where the lid is not fully open nor fully closed. So it hangs like this, and that shape basically allows it to get inside the minor. So that is why the H stereochemistry is very important, because we also did racemic ones, and the opposite Han stereochemistry, they lost the active. So this is one aspect. Then there's something which came as the dimers, one of the UK group, where the two scaffolds were brought in to certain spacers. You are looking into this. So this also went up to LJG 132, went up to phase 1 of the clinical trials, but again it had some bioavailability issues. So we looked into how to improve the bioavailability, and we brought in this type of a dimer a piperazine type of a moiety, which makes the molecule, because these are known for improving the bioavailability. So basically improve, so not only it improved the bioavailability, but it also improved the DNA binding. So how do you talk about this binding, the DNA ability, binding ability is? In a very simple terms, I would say that if you can compare this with just a simple of a coat or whatever it is, you consider that as the DNA. So when this molecule basically binds, so the zipper is like opening the zipper. So you do this at certain temperature. So that's how it's called as thermal denaturation of DNA. And when a compound is binding to DNA, the thermal denaturation of the DNA will be much higher. But if it's not binding, it's much lower. So once we get higher thermal denaturation, you would say that this is what is a much better DNA binding compound. So that's how you see that's what the examples what we have. This is basically, you can't see the results because of this strip. This is much lower compared to what we got these type of structures with the piperazine in the piperazine in the space. That's how such molecules were brought in. And I would say I will maybe try to take just 2-3 minutes to wind it up. I think that's okay, Chairman. A lot of other molecules are there. I want to just bring it to you. The molecule there. Something is about again. These are the couple of molecules. These were all licensed to a US company for their clinical development. So these are the 5 patent series, which were again US, BCT, all Indian, all patents. These were all licensed for their clinical development. So this is, I mean, this has taken at least, I would say, to bring it up to complete the discovery aspect to such series of compounds. It has taken us something like 4 to 5 years, because we were working on a large number of scaffolds. Then these are the part of us. So if somebody I could not, I mean, you have already understood, I will just straight away go to. What our group who had some of the students who had come. For I would say when I was aged 60 years, couple of there was a conference in our age. The building, what we call it as the Discovery Building. The name of this building is Discovery Building, where we have spent several years, and it so happened that quite a few had come from abroad, from within India, and so on. So this is, I mean, couple of them are, I mean, now today, most of them are either factory, they have their own industries. And also quite a few, I would say, are in very good academic positions, because it so happened that just last week, I mean, one of my former student who is in University of California, is the basically the additional director of this medicinal chemistry for anti-cancer drugs. And he received about $9 million of research grant. So I felt very happy. I mean, his training has really helped him get to such stages. So I think with this, I would like to thank you for all of, I mean, for your kind attention, although some of you may have been like some complex things happening with respect to these structures. So, because I was not aware that people from chemistry or pharmacy background may not be around much. So, anyway, I mean, at least it's good with respect to the students. I'm sure to get inspired, because as a chemical engineer, I will tell you, if you are into that also, there is a great opportunity working in office skills of human fluids and so on. And many of these things are a great inspiration. So thank you once again. If any specific doubt, anything, I'll be more than happy.
I think some pharmacy students are also here. Pharmacy scholars are here. In such case, I have some questions.
Please, please.
Sir, before you say that you are asking the Indian and A number, the pharmacy students, I believe you try to help us to find the problem where A number can be applied in the pharmacy. Can we make use of the drug delivery system? Can we make use of
We can use in many things. I mean, from discovery to their delivery aspects, many others. So many predictions can be done. But the only thing is, as I told, large number of pharmaceutical companies are already working on that. And but we never know, because nothing has come out practical. So how far it will be successful, we don't have any gold standards for that much of it.
I have another general question, sir.
Okay.
People say that the generic medicines can substitute some of the medicines. Generic medicines can substitute some of the medicines that are available in the market.
Yes.
And because let us say it's a open platform where commercialization is not there. Can we make use of generic medicines in place of the other medicines?
Why not?
Yeah.
Yes. There's nothing wrong with that. Because as I told you, just like a basic study, there is a little psychologically very different that medicine from a very reputed multinational would be good compared to what that also generic. It's not bad. They are also good. I must only tell you, the medicines what sometimes you will get the market, if they are adulterated, it's wrong. But the ones which are generic or the trade name, trade branded ones are exactly same. There's nothing wrong with that.
Thank you very much, sir. We are very happy for your presence, for your lecture, for inspiring us. And He has predicted one point. The medicine can give the side effects. Well, I will tell you my own example, including the name of the medicine also. I use the Metexel 50 for the BP.
Oh.
And that Metexel 50 gives the impact for the brain depression. And
And
then I went to another doctor. Then he asked me,
Link
don't
first,
make use of a
Video
Metexel
flight.
50. In place of that, you make use of a Conclave 5 and that gives less impact than the Metexel 50. And that way, you must be very careful. Even the doctor prescribes, you have to go to another doctor and take the opinion of another doctor also. Otherwise, you will be in problem. And his lecture has inspired me that way also. Thank you very much. Sir.
టచ్ కాదు కదా ఇది జస్ట్ నా ఎస్ చేస్తాను షేరింగ్ చేయండి చేస్తాను
To have another expert. I think this should have the first speaker. The lecture is on why should English use biology. Why should engineers use or why should engineers know biology? And really this particular title of the particular lecture itself has impressed me. The reason is when AICT has prescribed the syllabus in the year 2019, they included the biology as one of the subjects in engineering curriculum. Then I fought with Professor Shahadar directly. Because at the time I was a vice chancellor. So I directly talked to him, sir, why biology is required? We don't want biology. We are engineering curriculum. Why biology is required? That was my response. Later I came to know why biology is required. What is the importance of the biology in the engineering curriculum? Even today I am telling you, the artificial neural networks which we are making in India are biologically inspired. And whatever the medical technology recently I went to MedTech, in Visakhapatnam, and I found many industries which have been established are biologically inspired. And thereby I said that the biology is the most important subject to be introduced in our engineering curriculum in the first semester. And I'm very happy for Dr. C.H. Mohan's project. He is a professor in the Department of Biological Sciences, which Pilani Hyderabad campus, and former director of CCMB. And I'm very happy for his presence. I think it will be very beautiful. Dr. C.H. Mohan is one of the India's leading scientists in the fields of molecular biology, biophysics, and biomedical innovation. He served as the director of Center for Cellular and Molecular Biology, Hyderabad, and later as a CSAR distinguished scientist in 2018 to 23. He is currently senior professor emeritus and head of the biological sciences at BITS Milan Hyderabad campus. He is widely recognized for his pioneering research on molecular chapriores and heat shock proteins and their roles in health and disease. His interdisciplinary work combines biophysical, molecular biological, and cell biological approaches to address problems of biomedical importance. Among his major innovations is a DNA-based diagnostic chip developed by his team. Let the people know about you, sir. A great personality. And commercializing through Saigon Diagnostics Limited. The technology received international designation as product of the year in 2008 for addressing critical diagnostic needs. He also developed an advanced condition response to nanoparticle drug delivery system for fungal keratitis capable of targeting infection sites and releasing medication according to disease severity. He has also served in leadership roles in many scientific organizations, including the Society of Biological Chemists, the Indian Biological Society, and the Andhra Pradesh and Telangana Academies of Science. Beyond research and administration, Dr. Mohan Rao is deeply committed to science communication popularization, inspiring students and the public through lectures, research, outreach activities, and educational initiatives. Thank you, sir, for your presence. We are really inspired by your presence and encourage you to be your achievements, but by the end. Thank you.
Now just before I came, there were two lectures of very hardcore science subjects. I'm going to be very light. I'm going to talk very general aspects. In addition, I have to tell you two things before. I was expecting, since I am coming to JNTUK, I was expecting engineering college students. So I wanted to tell them why biology is important for engineering, because the discussion is very critical now. But now there's lots of research students and research faculty and other people are there. What I prepared for is actually engineering students, small level people. So for you it may be very breeze like, but good that you could relax in that. The second point I want to make is that I know whenever I leave Hyderabad and go to some other places to give lectures, there will be definitely some technical problem about presentation. So I carry my laptop. My laptop has a C type of output. So I carry my adapter for HDMI. I carry my cell changer. I carry my laser pointer. Because I know everywhere there is something. But today now I came here. I could not use any one of them because there are lots of complicated connections are there downstairs. And I hope I'll be able to go through my presentation. The point was mentioned that why engineering students should know biology. So we just start generally and talk about it. Yeah. So this is the picture. This is the picture of a bullet train. I'm sure all of you have heard of bullet train. This bullet train was launched in 1964. This is a launch picture. This is Japanese, of course, Japanese person. And they were launching the train first time in 1964. As you know, bullet trains are very, very fast trains. Because it's very fast train, when it enters the tunnels, when it is entering a tunnel, there is some problem. What type problem? Tunnel is like this. When tunnel is like this, the train is going in, it's like a piston. It's like a piston in a syringe. Because you don't guide like a piston, it will pressurize the air. It will pressurize the air, causes problems. So that's how train will be there of these moves. No. Can just present back. Let's check.
ప్లే కూడా ఉంది అక్కడ ఉందా
So there are many. Okay, this is the problem. It's engineering college, technology college, cooling technology. Very sorry. Now, here what we are showing is that the train moves. The train moves like this. This air here and train moving is pressurized. Because it's pressurized, then the resistance. If there is a drag, because of the drag, train will not go fast, drag is there, and then when it exits, the video is not there, but when it exits here, there is a sonic boom. Because it's coming out, all the air will come out and sonic boom. Now this will reduce the bullet train speed. The bullet train was not going more than 150, 170 miles at a time. Now they need a solution. Now what solution they find? This is the solution. This is a kingfisher. Kingfisher bird. The kingfisher bird comes from very high distance. Suddenly comes into the water. When a kingfisher knows beak is going to the water, you see there's water not splashing. There's no water splash at all. It just goes straight. Like immediately it goes in. Now there is no sonic bomb here. There is no disturbance of water. So if you take a kingfisher nose, beak, the train must go without any problem. So that's what is the idea. And engineers have drawn this. Just see the beak picture and draw the train diagram. And now this one made. This is the bullet train. Now, now the we are in biology. The biology is necessary for designing a train nose. Like that we can go. Now this is the this is a big what is a wind fan that is rotating. Wind is not strong all the time. Wind is sometimes strong, sometimes fast. But wind is erratic. This may not continuously move, and something not stall. It will stall up, it will stop, it will stop. The stall must be prevented. How do we prevent the stall? This is again drawback example. There is something called humpback whale. This whale has a hump here. This on the back of it is a hump. So that's why it's called humpback. This humpback whale on the fins, there are some structures here, small structures. These structures help it to go very fast without any difficulty. Now, once you will see that, now this is what can be done. See, because of that, because some small things of the structures are there, these structures help eddy currents locally, and those currents will prevent fog. So now you design a pin pipe. You put dots like this on the fan, it will not stop. So start will be delayed. So now you can design windmill using the pump wheel. Now there is another engineering problem. Turbulent boundary layers and drag, especially at high speeds, lead to lower speeds and higher fuel efficiency. If you have a very fast car, or airplane is going very fast, on the skin of the vehicle, or the surface of the car, air is touching. That will create a friction. There will be drag. The drag will cause fuel loss. Now, what is the biological solution for that? Sharp skin. Now we are saying birds. Now this is for shark. Shark skin has tiny ring-like tentacles that reduce flow of separation and turbulence. Now we can use the engineers develop ring-like pattern and gimmick that mimic the shark skin pattern. Pattern topography can be changed so that these machines can run faster. Now that's a design. So that's a shark skin. And if you look at the skin, it is like this. Scales are there. Its structure is like this. This is called tentacles. This kind of structures you can use on airplane surface, car, gas turbines, and wind turbines. So now again you see how biological knowledge can be utilized here. One of the beautiful things I think people should know is that India is making lots of everything aircraft we are able to make, but our aircraft engine is not available. India doesn't make aircraft engines. We have to import. It has a turbine, the turbojet. That turbojet technology is so difficult that we are not able to make. Now these are the technologies which are needed for turbojet design. Now that's the reason why you should know biology insights into engineering projects. Now this is another very beautiful example. I hope it not, but most likely unlikely. Now this is the biological example. This is lotus leaf. On lotus leaf, if you put a drop of water, everybody knows water will not touch the lotus leaf. And we've been using this idiom for long time. Now, why is that? Why is lotus leaf like that? Or biology people know there's a waxy layer on the leaf. In addition to that, somebody went and looked at the electron microscope picture of the leaf. Look at the electron microscope picture of leaf. You will see slight bud-like structures. These bud-like are nano structures standing erect on the leaf or small nano pillars are there. Because of nano pillars, water is not able to touch the surface. It will fill up air. And what does it touch? It is sit on top. When you sit on top, contact angle becomes more than 150. When contact angle becomes more than 150 or more than 120, water will not be able to stay. Okay? That is the main idea. Now, using this idea, one can make something nice, but I don't think it will not do. But tell me still. No.
Not saying, sir. Videos.
No, I have 7 videos. A new system will not take any video. I had laptop, but anyway, it is not connected. This shows what I'm here. This is a Korean piping bed. This is a bottom surface here. You take a glass surface, on it you put a narrow rods, the way we had in lotus leaf, exactly like that. Then put a drop of water and throw a drop of water. When you throw the drop of water, it will not stick, it will bounce like a ball. It will bounce like a tennis ball. That's the video picture that I had. Here the video was there. Water keeps on bouncing up and down. Now that's a beautiful, it's called super hydroponic surface. If you make a super hydroponic surface, it can be This is lotus leaf. And artificially made narrow structure. But what's the use of it? Your car windshield, when you are driving in the rain, wiper your wiper does never gets treated properly. If the wiper, if the windshield is made like this, water will not stick at all. You can build outer building coating can be made with this, so rain water will not spoil the building. Internally, organ, if you are implanting any organ, any material inside the body, that doesn't get touched with any of the skin. It doesn't get spoiled by anything. You can do lots of things with this material. This is bio-inspired, lotus leaf inspired, super hydrophobic structure with a nano direction. This is what one can do in the bio. Now, okay, this is a simple example. Now, this side of the picture is taken with a slightly cheaper camera. This is taken with a better camera. The difference is many people know HDR, high dynamic range. Okay. Now, high dynamic range is the lowest intensity, but the highest intensity, what is the ratio that we can do it? Contrast. So lowest to highest. Now, this is the best camera, this is the ordinary camera. But human eye can look at as large as 10⁻⁻6 candles per second per square meter. 10⁻⁻6 candles per square meter. It can go up to 10⁻6 candles per square meter. Just imagine 10⁻6, 10⁻3 is 1,000, is million. Million candles in 1 square meter, that much of light we can see at the same time we can see 10⁻6 times. We can see all of them at the same time. So that's the beauty of our human eye. And if you are actually in the darkness, completely closed, or fully darkness and you close eyes, and after some time you open your eyes, you can see single photon. Your eye capacity is, you can single photon can be seen. So eye is a fantastically high dynamic range equipment. Our heart from 7,600 liters per day. 7,600 liters per day, non-stop. Since the work, it is continuously pumping, it never stops. It stops at the end of the story. So this never stops.
With 7,600 liters, all ages or with age?
It changes a little bit as a person because the heart size does not change much. Heart size is this much, it slightly increase. It doesn't increase much. How the body increases, but because heart is pumping, the volume that are the 4 chambers, those 4 chambers size will not change much. So this will be around, let's say, 6,500 to 7,600. Not doesn't change quite. Then our body length of the blood vessels in the body are something like 96,000. 96,000 kilometers. 96,000 kilometers of blood vessels are in your body. From the blood is getting pumped this much every day without stop. So that's a massive engineering aspect. Then our human skeleton. Since I don't have enough time, I'm not going to details. Human skeleton is fantastic organ. You can jump from one place to another place. You can jump on your feet. The number of area of this size and your body weight if you count, that's a massively high energy. Now that is not easy for anybody to duplicate that. For example, when artificial legs are being designed, this is one problem. You put a step here, how much weight you are putting on a small area. So that has to be calculated and what bone will take care of. Bone actually has a beautiful structure. Outside is hard, inside is soft. Inside soft also mineralized, brings calcium, brings tissue, and not makes it hard. So internally always mentally hard. So it's a beautiful structure. Then human brain. Human brain is again fantastic. There's so many areas of curiosity. IBM Summit is a very fast computer. Now second fast. After the last time was the faster. IBM Summit is a supercomputer controls 30 megawatts of power. The power consumption is 30 megawatts. 30 megawatts almost like one power station production. Then it is capable of 200 petaflops. The operation petaflops you know floating point operation per second. Peta is 10⁻15. So this computer can do 10⁻15 floating operation per second. Human brain can approximately do 1 exaflop. 1 exaflop is 10⁻18 flops. So this is 3 order of magnitude higher than the capacity, but that is not the point. Because now already one supercomputer has come last year, which is faster than this. That's not the point. The point here is this needs 30 megawatts. This needs 20 watts. Okay. One roti, one chapati is sufficient. Okay, but now this is 13 megawatts, so very, very powerful system that. Now this is a picture of a computer. When the computer engineers, if anybody is there, this is what India. This was built in 1946. India electronic numerical integrator and calculator. This system was 30-ton machine. The machine was 30 tons and was 18 feet high and about 80 feet long. And this was having 17,000 vacuum tubes. I don't know, many of you might have forgotten, earlier we used to have vacuum tubes, then transistor, then your chips have done. So this vacuum tubes are 70,000. And that capacity was 1 lakh operations per second. 1 lakh operations per second was its capacity. That can considered very, very powerful. That was the computer. And that computer apparently when it started, it was in New York. When it started, New York City power supply was fluctuating because it is drawing all the power. That was the computer, it can do 1 lakh operations per second. Then IBM mainframe systems have come. Then tabletop computers have come. Laptops have come. Now our cell phones. These cell phones are so powerful that now Snapdragon 8, then 5th generation one is going up to 4.2 to 4.6 gigahertz. Now it's actually it means jigs, it used to be counted as jig, 0 instruction per second. We are calling blips, blips. Now it is jigs, giga instruction per second. He is in a cell phone. In a cell phone which is 30-ton computer and this cell phone. So that's the difference in size that has come. Now, very interesting engineering question is how small we can become. Now, 30-ton computer because smaller and smaller and smaller came to your farm. How small it will go? Will it go further small? If it goes, how small it can go? That's a unique question we should start thinking about it. It probably cannot go much smaller. This is one chip. This is actually 741. I use 741 long time ago, so I picked up. Now these are the circuits in it. You can see the connection. These are basic connections. These are internal wiring. This is one chip. Now we go to another chip. Today's chips. This again is a video. This is a video which scanning electromagnetic picture of a IC of a processor. Take a processor and go through scanning electromagnetic as deep as you go. This is a video. This video goes up to 1,000 angstroms. 1,000 angstroms it goes to very small curve. So now we are building chips which are very close by. So why is that? Are coming closer. If you make any more smaller, problem will come. What problem will come? Let's analyze. See, let's say this is one wire, one channel. This is one channel. This is another channel. In between, we have a insulator. Plastic, some silicon material is there. On which we are putting the tracks. This is one track, this is another track. It's very close. The tracks are like that. Insulator is there. Then current won't. There is a barrier. So the barrier, so electrons current won't pass. This is what is happening. We are making this smaller. This area, gray area, smaller and smaller and smaller. Right? Quantum smaller. It becomes quantum tunneling. Yeah, now it comes quantum tunneling will start coming. When quantum tunneling starts coming in, electrons will go through. Electrons will not stop this. Insulator will not be able to stop. When insulator will not be able to stop, electrons will go. When electron goes through, and then there is a function like it means we are going to have a limit of miniaturization. We will not be able to go continuously. We will have limit. That limit is called tunnel limit. Tunnel limit is there, of course. People are working on how to overcome tunnel limit. There are many methods of overcoming. Multi-layer chips, everything is coming up. In addition to that, one good way of overcoming this is molecular computation. There is a paper published last year, July 25. That paper says beyond silicon, a roadmap for overcoming challenges in molecular computing. Now, silicon-based computing is we slow down and start molecular computing. In molecular computation, this tunnel effect will not come into it because molecules will function differently. Now, currently we are using top-down approach. We take a material and make it small. This is somehow material integration. In molecular computation, we take molecules and put the molecules together and build a system which will be more efficient. If you look at the time process, 1955, our chips used to be about 1 centimeter long. And then become smaller and smaller. Transistors become smaller and smaller. Now become transistor 5 nanometer, 25. It can become smaller than now. Huh? 50, 50, yeah, this is 5 lines. So 5 nanometer also we can make it that. Today one can make use sub nano technique also. Now we are going to quantum computation, we are going to photon computation, and we are going to molecular computation. So these are the areas which are going to be in future. For that reason, you cannot stick to one area. You must know physics, chemistry, and biology. Then only you'll be able to build these things in future. Now, because this expansion is going on, you have to have a different understanding of biological molecules are very good molecules for molecular computation process. If you want to do molecular computation, biomolecules, what are biomolecules? DNA and protein. You can use DNA, protein, carbohydrate, lipid, any other thing you use. But DNA and protein are very good because they're polymers. They have some properties which we can use. These are excellent materials because they are complementarity for cell perspectives. To take DNA is double helical structure. If you pull the two strands separate, each strand is complementary to other one. So this complementarity can be used. And proteins are long polymers. These polymers can be utilized for different purposes. Biological molecules they can conduct, produce photovoltaic voltage, behave as molecular machines and exhibit photochromism properties. So with this can be utilized. So that's what DNA is double helical structure. And it probably can conduct electricity. If you take a DNA, apply plus and minus voltage, it probably will conduct. Initially, not suspected will conduct or not. But when the experiments were done, it was conducting, and then the people had it out. Because all biological molecules are in buffer solution. You take a buffer solution and dry it and put it there, buffer salts will be there. So these salts may be conducting. The salts are conducting, DNA is conducting the other. Now, but now we know that DNA can conduct it. It not only conduct electricity, it conducts electricity depending on sequence. When you say sequence, DNA, as some of you know, the long molecule which has 4 nucleotides, A, T, G, C. Adenine, guanine, cytosine, guanine, these are all 4 different bases. That in a word, each alphabets are there. Alphabets make a word, and words make sentences. Like that, by changing alphabet position, you can change the meaning of a DNA. Now, sequence can be changed. If one sequence is there, or GC, GC, GC is there, this will conduct differently compared to all TT, AT, conduction will be different. If that's the case, it would be possible to build a diode. You have a P type and N type structure. You have one sequence here, that's work like a P type, N type diode. So it should be possible to build diodes to the molecular components. Now DNA can also come together, form something called holiday junctions. Okay? These junctions can be made to scaffolds. Means you can make a patterns. DNA patterns can be made. Now proteins, if you leave proteins, they can aggregate. These are specific proteins. These are the organic proteins I worked on in my lab. These proteins can come together, form like a long structure. This kind of a long structure can be formed. These are fibril or a length of some kind of a wire. Now, I think probably this may show up. Each spot is some kind of protein in a fluorescent microscope. If I allow this protein in some other condition, special condition, they can attach each other and become a long fiber. Now, yeah, this is now you can see that 15 minutes, 30 minutes, you can see some of them are becoming longer. Here, they become long. So 180 minutes I have. This one is becoming long, this is becoming long. So this becomes long. If you leave, these proteins are becoming long. Long, but it is still nanometer scale, very small scale, they become long. Now this can be probably used as a conductors. So for example, you take, I think many of engineers know beam technique. They just make a film first, put a polymer and put an electron beam here and make a vapor, all these things. By doing all these things, you'll be able to generate. This is a metal piece, this is a metal piece. There's a gap in between. If you apply voltage to across these plates, it will not conduct because there's nothing in between. But if you put a protein fiber here, put a protein fiber series, black ones are fibers, put it up. You put that, you measure the voltage. As you increase the voltage, current will increase. This means it's conduct. So you can make a nano protein wire. Now, this protein is a protein, same protein. In one shape, one conformation called beta shape conflict, one called alternative conflict. Same protein will change the conflict. In this conflict long, in this conflict it's short. Okay. If I put a plate here, electron plate here, and plate here, then you connect and make the switch on. If a conflict change, if you come down, break the circuit, so the system will be off. So by changing the confirmation, one should be able to generate a switch, a molecular switch. Now, similar to this, I have done good. This again movie, it doesn't move. Here, this is a molecule called ATP synthase. This molecule makes ATP in the body. Now, this is alpha subunit, it's alpha subunit, it's alpha subunit. This is beta, this beta, this green. Center of blue is a gamma. When ATP synthesis is happening, this gamma will rotate. Gamma will be rotating. Gamma subunit will be rotating every time in your cell, your body, in your cells, these molecules are rotating. Now, can we use this rotating molecule as a motor, dielectric motor? Fortunately, this you can see. You can see that. So here we are putting that those are subunits. On the top there is a small metal piece. That metal piece is rotating. Okay. So it should be possible to make a small motor out of these proteins. So what are we making? Proteins and molecular. These are one level. You can also use proteins, molecular electronics. Electronics, you can do electronic, mechanical. These proteins, which are proteins, some of them I work with, azulin, maltose binding protein, ATP synthase, protein fibers, all can be used in electronic. Rhodopsin. Rhodopsin is something which we have in our eyes. We see our vision is because of rhodopsin. Rhodopsin, phytochrome, what these molecules can be utilized. Now, one example is eye. All of you know this is eye. This is a lens, eye lens. Vision picture will be formed on the retina here. On the retina, there is a transducer, like CMOS chip. CMOS chip, you have your camera. So this is here. But instead of CMOS chip here molecules change. This molecule cis retinol. When the light falls, it becomes trans. Cis trans means this position here. You see, this is in light, this turned down. Here, line bent. So here line is not bent. This is cis continuously. This is bent. This is molecule bending happens because of light. When light is absorbed. Now this, because of that, we are able to see. Our vision is because of this small molecular change. Now, you can take rhodopsin from a bacteria and use it. If you use here, if you shine light, it will convert into something. It will process it. It releases a proton here and comes out and again picks up one proton and goes. It's releasing a proton and outside, picking a proton from inside. So if I am sitting on a fence, sitting on a fence, from one side I take a proton, one side I release. If you keep on doing what happens, more protons here, less protons here. That is potential difference. It creates EMF. That EMF is the one which we are using for seeing. Now, that's where I said the first light is absorbed, and that's where the proton comes out. Now, that part of the chain, if you look at it, only part if you look at it, there's an interesting part. Here, 640, this area, in this area, when the signal is coming here, if you shine certain light, it will go to it will branch out. Again, you shine there, it will come back. This is very stable. By using this, we can actually write. We can convert 0 or 1 bytes. At one level it's 0, another level 1. So bytes can be made like this. Now if you shine light, it will go through all processes and go to 640. At 640, if you give a light, it might put one bit here. Now if you erase it, give another light, it erases it. If you don't erase, it goes to this distance. So it means my rhodopsin molecule can be a memory unit. Like if it's 1 or 0, like 1 or 0, you can use this molecule for storing. Now, why is that important? If you take 1 ml of solution, there will be billions and billions of molecules. It means you will be storing billions and billions of bits. It means your gigabit storage capacity will be dramatically improved without electricity. Now, same thing can also be used as a logic gate. You can put one light here, then only one light here because the AND gate, the process will not happen. When both the lights are there, it happens. Of course, there's a lot of story here by what I have time to explain that detail. We can also use a photon light switching. In this, light is coming, laser light is coming, it's diverted here, coming here, diverted here, going. Here I have drops in molecule, here I have drops molecule. I can change the direction of light. Now this is going like I can switch the light by conformational change. If I do the conformational change, switch. Now we notice the light is coming from here, it's not going there. So it was here, it was here. So you can switch the light direction, switching direction by changing the conformational molecule by again photosensitive effects. So that is also possible. Now, if all these things possible, we are making wires, switches, motors, and gates. If you can make all these things, you must be able to build a robot. You can build a micro robot, a nano robot. Now, can such a nano robot be built? Long time ago, Rajat University made this video clip as a possibility. It did not happen, right? As a possibility, they made this. It's very interesting to see that we can build a nano robot. That nano robot will have a drug molecule. You inject it, it will go into the body. It's going to blood vessels. When it's going to blood vessels, if anything is bad, it will identify that which is bad and release the drug there. So only that gets destroyed. So that your drug effect side effects will be minimized. Now that's how that's a video again. There's a video where these blood cells are going through and a nano system goes inside. This idea is similar to Asimov's book, Fantastic Voyage. Has anybody read Asimov? You have to read, the students have to read science fiction. The Asimov wrote a science fiction novel, it's called Fantastic Voyage. That Fantastic Voyage is that one Russian spy is dying, but he has lots of influence in his mind. So people want to information out. There is no way because the person is dying. So what they do, they miniaturize the doctors. They miniaturize, it's a novel. They miniaturize the doctors and inject them doctors through the blood vessel. They will go through in a boat, small boats, and go through vessel, go to the brain, and then collect the information. That's a science fiction mode. Now what we are talking about is make a nano robot, inject, they will go inside and do the surgery. That's a science fiction. But don't worry, that will become possible. Sooner or later, you'll be able to make that possible. So first such now what we are doing for all these things to do that, we need molecular understanding. For molecular understanding, for such molecular device, we need to monitor biology. Why we need molecular biology? We can change DNA as we want. DNA can be tailor-made. If I change DNA, I can change protein. If I change my protein, I can change my conformation. So all building units are under control. We will be able to do whatever we want. So because of this, it must be possible to build nice things in future. So just biology 101 course is that all biology, our bodies, our beautiful bodies are made with the cells. Cells, this is one cell. Cells come together to form some kind of structure we call tissue. So cells will make tissue, the tissue will make organs. Organs is the heart, the liver, intestine, things like all these organs are made by tissue. Inside the cell, we have chromosomes. You must have heard. Inside the chromosomes, we have DNA. We have DNA and protein are inside. Now DNA is a double helical structure which we talked about. And then this again video which shows how DNA makes a protein. DNA makes RNA, RNA makes the protein. How this is made in cell. The 2 minutes video was a very beautiful video. This video was made by NIH scientists to convince the parliament and US Parliament, US Senate for funding to explain to people what is basic dogma biology. To tell the Senate members, they made a video. It means this video must be very, very simple for everybody to understand. That video, of course, is not big. This is the last couple of slides. This is where the young computer science students should pay attention. There is a protein that is isolated from jellyfish. Jellyfish, you know, I think you are in KAKINADA, so you must play going to beach and you saw jellyfish. All of you know jellyfish, no? Okay. Jellyfish, from jellyfish and protein isolated are GM, green fluorescent protein. That protein looks like this. It's called beta sheets of helix, so don't worry about that. These protein amino acids come together to form a structure called this pigment is formed. This pigment is green fluorescent. Now this is a very nice molecule. It was made long time ago. Person who made this won the Nobel Prize. Because this protein is fantastically useful for biological research. So this is very important. Why am I showing this protein here? Because very recently, last month, in January, sorry, not last month, January 2026, there's a paper in Nature. The Nature paper shows that this molecule, the GFE molecule, can be shine, you can shine light on it. So, you shine light, it will flow. It will be a flow that. What happens if you shine light, it goes to excited state, from excited to ground state. While it goes to ground state, it emits light. So it's like a photon, that goes. Sometimes from single state, it goes to triplet state. When it goes triplet state, again, chemistry guys, if you are there, so triplet to single state is not permitted, it's a forbidden transition, so it stays there. Now we have electrons, can you trap that in one state? Under those conditions, the excited electron and nearby electron in the molecule are entangled. Now you heard the word quantum entanglement. So these electrons are quantum entangled. When they are entangled, you can give radio frequency from outside. When you give radio frequency, these entangled electrons will come superposition. Quantum superposition. Have you heard the word quantum superposition? This quantum superposition happens when you give a RF to this molecule. First excite the molecule, both single state and different state. Under the content of radio frequency will make it as a superposition. When quantum superposition takes place, if I have any external magnetic field, this is what is shown here, excited state. Now you have charge boxes take place. This is the both states, 0 state and 1 state. There's a spin superposition state. Spin can be half plus half or minus half. Then can be superposition. This superposition situation is coming. Under these conditions, if there is a magnetic field nearby, the magnetic field nearby, the superposition gets go away. Again, you understand the quantum collapse will happen. So if a magnetic field is there, collapse will go away. If collapse goes away, flow intensity drops. Flow intensity drops, it comes back. So it is possible to detect magnetic field variations in using this technique. So this will be, that's what quantum light becomes, that's what it happens. Now, this is the basic point. A triplet state and a entanglement between two electrons in the ideal parts of the molecule. Magnetic field affect electron spin states, and this alters intensity of the fluorescent light, turning protein into useful quantum sensor. So this paper appeared in Nature, January 2026. Anyone of you please go and read the paper. It is a breakthrough. Why it is breakthrough? If you are following up anything about quantum computation, why is quantum computation so expensive? Only some countries are able to do it. Because the quantum states superposition is stable only at an extremely low temperature. So we need to have a very, very low temperature. To maintain the low temperature is very, very expensive process. So because of those situations, we are unable to develop. Now this protein acting like a qubit. Actually, I have no step. Now in normal case, you have a 0 and you have 1. Now it is 0 or 1. But in quantum qubit, it is 0 or 1 continuously. There is variation possibility. If you look at it, quantum superposition is possible. Now in the quantum world, we can intentionally make a superposition and control it. This is one of the main properties of the qubits. For qubit, we need all conditions. But this protein is a jellyfish protein. Jellyfish protein, you see water temperature is 37 degrees, or 27 degrees. So room temperature, jellyfish protein with a very noisy and destructive atmosphere, it is showing quantum bit properties. If that shows qubit properties, it means you don't need a low temperature. Now, this is what the quantum computer looks like. Quantum computer should be kept like this. Okay, it's a massive expenditure. Now, under this case, if a protein becomes qubit, so what if protein becomes qubit? If a protein can become a qubit, it means you don't need any one of them. If you don't need any one of them, suddenly quantum computation becomes affordable in everybody. That is what is likely. What if protein qubit become reality? Future is for interdisciplinary experts, the people who know both areas, biology, chemistry, and physics, and mathematics, and computer science. People who can understand many languages, so people who can understand many subjects are the ones who are going to pay. Now, technology experts with the domain knowledge. You are a technology expert with the domain knowledge. If you have this, you are unique compared to anybody can code. Millions of people can code. Coding everybody can do. Nowadays, AI will code to the coding. You don't need any people to code. So you don't need anybody. So unless you have a special brain is that you will be able to understand multiple things. Then and then only you have to. And if you want to make any difference in the world, you must learn multiple areas of technology and keep learning. And this is the reason why biology is important, because I showed you mechanical examples, engineering examples, quantum computer examples, and process example. And biology we can control everything. DNA sequence can be controlled, protein can be controlled. Because we can control these two, it means we will be able to fabricate anything that we want. So there is a good potential of molecular computation becoming reality. Protein qubits become reality. They are not real now. Today they are not real. But there is a possibility that they are becoming. And you young guys have a chance to do it. We have done whatever we could. I don't think we have enough time to do anything more. So it's not for you. Thank you.
I invite the students to interact. Questions, please.
Mike. You go to channel.
Thank you very much. We are inspired by the lecture. We are also picking up biology as one of the
Mike is gone.
regulations. And today see guidelines. We get your expertise.
Yeah. No. In many colleges, many institutions, biology is a compulsory for all engineering students. In physics, chemistry, and biology, please learn in the first semester or second semester for all engineering students. At least in which Pilari and IIT it is compulsory. Now we are introducing in Pilari that every student, whichever study subject is studying, at least 10% of his courses must be AI, must belong to AI. You're a biology student or a history student or economics student. You must study AI, 10% of your course must be AI. If you don't know AI, it means you don't know English. Now that's the situation. So we must learn that. So there is no alternative.
Yes.
We must introduce AI as a compulsory introduction course. Let them play, not ChatGPT. ChatGPT is a useless stuff. Everybody is a ChatGPT. It tells you all wrong things. I don't know whether you notice. The general collection of information is fine, but it makes many mistakes. I'll tell you one example. I was invited to go to Nepal. Nepal to give a lecture on modern science and evolutionary theory, Darwin's evolution theory. And this meeting was organized by Vedantic community. So Vedantic community, though they don't believe evolution took place, they think God made everything. So evolution is not correct. As a scientist, I have tell them that Darwin's evolution is correct. Okay. Now I am going to actually counter large number of people's opinion there. So I was preparing myself. So I wanted to start my lecture with praising ancient knowledge. So I wanted a shloka which describes science as important aspect in human thinking. Like ancient Indians were very good thinkers. So they had so many, they did mathematics, they did astronomy, they are fantastic thinkers. So is there any place, is there any shloka which describes ancient Indians' knowledge of analytical capacity or importance of analytical capacity? That's the question I asked. He gave me beautiful shloka. I was impressed with that. Then I said, okay, I am going to use this shloka for my lecture. I said, please give me the because I want to put the where is this so and so chapter. It says, no, so sorry, no, sir. Based on your prompt, I made this shloka. Okay, now, what do I do? If I take the shloka and I showed it to the Vedantic people, they would have thrown me out. Okay, so I think ChatGPT, if you're using, be careful. ChatGPT, use ChatGPT for searching, but use your intelligence to make other conclusion. Otherwise, you'll make a mess. All these AI programs, what AI can do, it can give you information on what is existing knowledge, the knowledge which we have given, and it's analyzing, creating something. So unless we use our own AI, it won't work. Okay. Yeah. But before closing, I want to tell the students, if their students are there, you must interact with the class. If you are civil engineer, up to electronic engineer, if you are electronic engineer, up to computer engineer, then your class population is very important for success.
Thank you, sir. Thank you very much. Thank you. We are very happy for the 3 lectures, and heartfully we invite their presence in future too at JNTUK KAKINADA to enlighten the students and the scholars. Thank you very much for giving me the opportunity. I thank the organizers for giving me the opportunity to be with you. Thank you.
Thank you, sir, Srinivas Magar. Thank you very much for chairing this session. One point I want to share with Mohan Rao garu. Correctly you told, sir. This one you mentioned one point. ChatGPT, right? With respect to one point, maybe a lot of our friends experienced it. Whenever we search at Google, it says something not available. But ChatGPT never says not available. Whatever, even the trash it gives. Okay, that's why I am accepting, coinciding with your statement. Now, I sincerely request our Professor KVSG Murali Krishna garu, Honorable Vice Chancellor, Vice Chancellor of the UK Kakinada, and Professor of Civil Engineering, Senior Professor of Civil Engineering, to chair this session. Session theme is Interdisciplinary Frontiers in Agriculture, Food, and Life Sciences. Please, sir, I will ask one of the volunteers to introduce the first edition. Now, the distinguished lecture in memory of Bharat Ratna, Dr. M.S. Swaminathan, Dr. C.H. Srinivas Rao, Director, IARI, ICAR, New Delhi, on Green Revolution in India, Technology, Synergy, and Strategy of Professor M.S. Swaminathan. Please give me 2 minutes time, sir, to introduce you to the audience. Please come on to the dais.
Good evening, everyone. It is my privilege to introduce Dr. C.S. Srinivasarao, one of India's most distinguished agricultural scientists and the current director of Indian Agricultural Research Institute, serving since December 26, 2020. He completed his BSc and MSc in Agriculture from Acharya N.G. Ranga Agricultural University and earned his PhD from Indian Agricultural Research Institute and pursued postdoctoral research at Tel Aviv University, Israel. With over 34 years of experience, he has held several prestigious leadership positions, including Director of National Academy of Agricultural Research Management and Central Research Institute for Dryland Agriculture. His contribution to agricultural science, climate change research, and sustainable farming has been remarkable. He received 49 national and international awards, including the prestigious Rafi Ahmad Kidwa Award and the Basnik Award. Under his leadership, institutions received the Sardar Patel Outstanding ICAR Institution Award twice. His academic achievements are extraordinary, with 363 research papers, 58 books, and 19 copyrights to his credit. He has represented India globally in climate change negotiations and scientific forums across nearly 20 countries. His journey, the true inspiration, showing us how dedication, knowledge, and vision can transform agriculture and contribute to national development. We are truly honored to have such a distinguished expert among us today. Please join me in warmly welcoming Dr. C.S. Srinivasarao. Welcome.
Before I invite the speaker, let me just share a few thoughts. We all are aware about the greatest green revolution, father of green revolution, Dr. Norman Borlaug. It is believed that his statues are found in almost all countries of the world. And he is father of green revolution in India is Dr. Swaminathan. And when this university was formed in 2008, we invited Sri Ambi Rao to the same place where Sal is sitting. He was he got Padma Shri also, and he worked with Dr. M.S. Swaminathan for nearly 30 years, and he died in the year 2016, 10 years. So he also worked with the Indian Agricultural Research Institute as deputy director. He was the vice chancellor of N.G. Ranga University, and we had the privilege to listen to that great person earlier. Now we are very happy that you are with us. and I welcome you all to give your opinion.
Good afternoon, everyone, and it's a great privilege to be here. Thank you very much, Organizing Secretary and JNTU KAKINADA authorities to make me part of this important event. So let me take forward how the story of the Green Revolution in India. As Honorable Chairman said, that is the father of Green Revolution, Bharat Award. I am from, of course, Dr. M.S. Swarnandhra was our former director, and the organization was established in 1905, 122 years old, and which was originally like this. And in fact, some of the conferences at that time was organized under this split. This is the current library of IRI, where it was named after Professor M.S. Swaminathan. And the legacy continues that how the Indian storms were there, 100 years celebrations were there of IRI. Let me take forward the how M.S. Swaminathan sir is so unique and in India. And we all know, dear students, in India, 7 lakh villages are there, and 14 crore farmers are there. So how the Green Revolution revolutionized the Indian agriculture and food system in the rural economy. We all know that over the centuries of India, the list of major famines goes in several, in fact, several the Great Bengal famine, Madras famine, and several famines which we lost so many lives. The Bengal famine of the 1943 killed more than 3 million people in eastern India. So these are the state of Bengal famine, where many children, women, and men died out of hunger. Taking the two of these things, how to handle the food systems in India, the Lal Bahadur Shastriji, then Prime Minister of India, requested that they skip one meal at least in a week time, so that others will have the food. And before telling, in fact, he himself was following it, then he advocated. At the time that the famines in the different countries, mostly US was supplying food systems. Then there were two. Can you remove this? Excuse me. Can you remove this? Minimize. At that time, you can see that the US can't fill the food gap for the developing countries. So that was the indication which I have made. That, for example, Egypt can't be saved. For example, Libya, walking wounded. India was named can't be saved. So that was the stage where India food crisis, food systems are happening that time. So Indian food price management is an inspiring science story. It has science and technology, it has policies, it has programs, it is a thinking process, it has philosophy, a culmination of all the Indian Green Revolution. The story early effects of the developing fertilizer responsive varieties were done in 1950s. The transfer of genes for fertilizer response from Japonica to Indica rice varieties that was Dr. Ramaya ji at CRRI Katak, he led that. And semi-dwarf and fertilizer responsive varieties of wheat using multiple research strategies, including radiation-induced mutant development. But unfortunately, in early efforts, what happened that the semi-dwarf strains had also reduced the conical size and which eventually reduced the yield levels. In prior to 1968, you can see that what type 1 ton per hectare. Hectare means 2.5 acres. So such a low yields because of tall plant feature and also thin straw. So which is susceptible to lodging. Whenever there is high wind speed, cyclones and these things are happening. So it is a lodging effect is happening. And less response to fertilizers and irrigation. But the same 1 ton, you can see that in the bottom wheat production, India is number 2, whereas the rice production now currently it is number 1 in the world. Then followed by origin of semi-dwarf wheat story in 1935. Originally, the JNTUK crossed a semi-dwarf Japanese wheat landrace with the 2 American varieties. That is a critical aspect happened. Norin 10 has been evolved with that. Then, what is the uniqueness of Norin 10 is that it's taller than 150. Traditional varieties are taller than 150 centimeters, whereas Norin 10 is reduced to almost 50% height. Then, taking the clue of that success, Dr. Vogel at Washington State University used dirty Norin as a basic material to help and produce high-yielding semi-dwarf winter wheat varieties. Taking that, the father of green revolution, as Honorable Chairman said, that he has used that Vogel's semi-dwarf varieties, which the previous one has developed, with the Mexican varieties to develop 2 important wheat varieties, Pitic 62 and Penjabo 62. These are the 2 varieties which are short straws and produce delivery, more grain per head, non-logic. They are which we want all farming community and scientists to want. All four features were evolved in this. Then taking the story and introduction of those varieties in India in 1961, semi-dwarf materials of International Right Trust Nursery, which were collaborated where Dr. M.V. Rao gave the story of M.V. Rao and his associate M.S. Swarnandhra service collaborated. Next 2 years, introduction of identification and new plant tags were developed. In March 1963, visit of Normal Borla. In fact, the association between M.S. Swaminathan ji and Normal Borla, that collaboration have really needed so much so that India has become priority country for Normal Borla. Then in March 63, visit of Dr. Norman Borla, extensive multi-location trials of semi-dwarf bean selections were taken. He visited across the India and at that time, after any time, convincing people, bringing them into the any science and technology has to be scaled up makes that political support is very much required. It has to enter the national programs and policy. Then only you can take the expansion. Otherwise, it will restrict a few villages, few research stations. Then September 1963, 100 kg of these improved varieties were brought. Then subsequently, then in 1964, large number of across India in several the multiplication farm speeds yields improved 1 ton to 4 to 5 tons of wheat. Then you can see that and the standard along with its economic means better soil management, better water management and plant protection, other things were added to the varietal strength, then a synergy is happened. 1965 to 66 selection of amber grain material, Kalyan Sona and Sonalika, that is a big success story in overall wheat production. And once varieties are developed, they have limited. So, as I was telling that it has to be, seed should be made available to the farmers in remote areas. At the time, these transportation seed systems were not prevalent. Then there are two strategies for farmers. One strategy is organizing wheat seed village concept. Seed village means that we adopt the villages and we multiply with the help of all technical aspects and the farmers field seeds are developed. Second one was importing large quantities from Mexico, 200 tons of Lerma Rosso in 1965, then followed by higher amount, 18 tons were in 1960 was imported. You see that the 4,000 years of achievement in production condense to 4 years. You can see in 1947-48, the wheat production in India was 6 million tons. In 1963 to 64, 10 million tons. Within 4 years, it increased to 17 million tons. The green innovation is that the MS Swaminathan sir, he told that development of improved technology alone cannot help farmers grow food unless the diffusion of the technology facilitates the public policy. Therefore, public policy with a strong technical knowledge of the researchers, policy makers, and the farmer participation is very critical. These are some of the power of scientific partnership which Professor MS Swaminathan adopted in that he has long liaison work with the foreign leaders. You can see that Norman Borlaug visiting wheat fields. Then power of the policy advocacy, he has convinced the political people. Here you can see that Lal Bahadur Shastri Swaminathan explained the significance of wheats to Prime Minister Lal Bahadur Shastri during visit to IRI that expected import of large quantities of wheat seed from Mexico. Then it is not only scientist success story, it is a farmer success story. You can see that the green revolution backbone has happened in Punjab farmers' fields. In fact, every Punjab, not only farmers, even old and young people, some of the people, everybody has strongly supported. If we see that the enthusiasm, hardworking, and skilled and determined Punjab farmer has been the backbone of the green revolution. Revolutions are usually associated with the young, but in the revolution, this revolution, age has no obstacle to the participation. Farmers, young and old, educated and uneducated, have easily taken to the new agronomy. It has been heartwarming to see young college graduates, retired officials, ex-army and illiterate peasants and small farmers keen to get the new seeds. So that was the story, because we have seen the food crisis in India and taking so much sorrowness and empathy that leads that the Punjab farmers to move forward. So that's green generation was coined like that. New opportunities for increasing production through a substantial rise in the productivity of croplands. These and other developments in the fields of agriculture contain the makings of new revolution. It's not a violent red revolution like that Soviets, nor it is a white revolution like that of a Shah of Iran. I call it is a green revolution. It happened in direct USAID. So this is the overall green revolution success story, which made when we were not having enough food, we were looking at as a begging bowl. The ship comes to some of the Indian territory. We are waiting to fill our bellies. But now we can see that 70 crores of Indians are taking the advantage Food Security Act 2030. And around 15 countries, India is supporting food aid. And currently agriculture GDP is 19%. And to cover 7 lakh villages, 14 crore farmers and 15 crore farmlands. This also led to the hunger reduction and poverty reduction. You can see substantial when agriculture growth happened, substantial poverty reduction happened in India. Then many additional attributes were coming to the society. This is at IRI, the celebration happened with the wheat production increased in 4 years. The stamp was released and Honorable then Prime Minister Indira Gandhi ji was at IRI, the typical symbol of synergy between technology and public policy. There are different phases of IRI leadership which has contributed in 100 to 1930, early days of handling genetics, where Mohan Rao Saab has explained that how biologists, engineers need to understand biology. A fascinated presentation. Thank you very much, Mohan Rao ji. And enlarging the base of theory to application. And the next phase is the green revolution. Now the gene revolution, which is happening in fact in that. Recently, IRI has developed first ever genome edited rice variety in the world. Then these are it's not only variety. The MS Swarnandhra is philosophy how technical integration. Usually in any scientist, also agriculture scientist, if any subject, if it is any soil scientist, it is a entomologist, any subject is there, he will try to demonstrate his own achievements in the farmer's field. But here is the strategy for the success story that he culminated all the better technologies and single field along with improved variety that made resulted in the miracle results. You can see that the rice and wheat varieties along the fertilizers, along with irrigation, along with other management practices, that really revolutionized within a short time. Then taking these are the key factors, seed sector, fertilizer, nutrients, pesticides. These are the 3 important inputs which have contributed to green revolution. And overall, the seed, combination of irrigation, fertilizers, pesticides, and mechanization, it is called that these are the green revolution components of this. The slide which I would like to express all of you, maybe many of you are the engineers, few biologists, few agriculture scientists. In India, you can see that the area under cultivation has not increased. You can see that the area is approximately say 142 million hectares, almost during last 70 to 75 years. You can see that the productivity has improved like anything and through various revolutions of agriculture. We are talking about green revolution to improve the food rights, rice and wheat and maize. And second one is the milk is a white revolution, oil seeds, yellow revolution, blue revolution is a fishery, silver revolution is the eggs, and the golden revolution is fruits, vegetables, and flowers. This is the which one I was highlighting that the blue line was stable, food production is increasing. The science and technology contribution, which can see that how the average yield has increased from 0.7 to tons to 2.4, 3 times more yields. At present, dear colleagues, India has 358 million tons of food grains. We have 360 million tons of fruits and vegetables, 240 million liters of milk, 110 billions of eggs, 18 million tons of fish. But we have problem where to keep and store rice and wheat. Just 70, 80 years when begging about, you have seen the Bengal famine where people were dying. Now the system is that where to keep that person. So that was the Indian success story where productions levels. You can proudly say that India, number 1 is rice production in India, second largest of wheat, fruits, vegetables, sugarcane, and record production of food grains, and the world largest producer of milk, pulses, tea, spices, jute, banana. The agriculture sector is one of the fast growing sector. Then we are exporting at present 65 billion US dollars of agri exports from India to other countries. And as a professor, MS Swarnandhra sir also contributed to IRI, different facilities, very foresighted infrastructure. And internationally also, as a director of IRI, then he has become secretary there. Then he is a acting deputy chairman, a member of the Environment, Women's and Development NBB, then Director General of International Rice Research at IRRI, then chairperson of the Farmers Commission, National Commission on Farmers, then Rajya Sabha member. This picture also shows that how many honorary delegate doctorates across and across the world, and also World Food Prize winner. He was selected. And he also made a foundation for basmati rice. Whoever is eating biryani, that originated from IRA. 95% of basmati rice is produced, and breeder seed is produced from IRA. And you can see also Professor MS Swaminathan visionary. He has not only made that one science, but he has also done that after varieties, food security was stabilized, and also he has also created that how many scientists are required for Indian Council of Agriculture Research. Now we are enjoying 6,000 agriculture scientists in India. This was originally created by MS Swaminathan. Once a scientist joined after MSc, PhD as a scientist, he started National Academy of Agriculture Research to transform the students into the thoughtful and young scientists. And how to recruit these 6,000 scientists? He has established that just like UPSC, ACE agriculture scientist recruitment was established. Then DARE was established. We all think that whichever district you are going, there are 2 KVKs, Krishi Vijnana Kendras are there. At present in India, 732 Krishi Vijnana Kendras are there. These were established by MS Swaminathan sir. So these are some of the World Prize winners, including MS Swaminathan. And he also thought that we have INSA, NASI, Bangalore Academy. We have National Academy of Agricultural Sciences. In fact, he was founder, president of National Academy of Agricultural Sciences. Then he has also coined how lab to land, green evolution, gender equity, seed community. But at the same time, the most important legacy of the MS Swaminathan ji is that, okay, we were starving. We need a technology. We need more food production. Fertilizer was being applied more, and also a lot of pesticides. Irrigation was taken intensively being used. Then at the same time, we also cautioned that what are the side effects of green revolution. And that's why he find that if conservation of natural resources goes wrong, nothing else will work right. For example, intensive cultivation. When Indian farmers were making one crop, I myself was the son of farmer, especially dryland farmer in Krishna district of Andhra Pradesh. Only one crop we used to take that. Then we increased to 2 crops, we increased to 3 crops. Now some of the states farmers are out to take 4 crops. So more crops means more fertilizer, more water, more mining, more the consequences. That's what he cautioned in 1940, 1960s, 70s. What are the side effects of green revolution? Groundwater depletion, chemical usage, soil health deterioration, lots of biodiversity, and climate change. But everybody, whatever said on that, I think everybody needs food. Today also you must have IT inauguration, have very nice lunch, and also afternoon. And again, after these plenary lectures, again. But anything can wait, but food cannot be. In fact, everybody expects that right time and quality and nice food. We have in the 8.2 billion world population, India is reaching 150 billion. Every year, 1.5 crores are added to Indian population. Therefore, if we want to see that agriculture sustainability, when we topic of the seminar is the Vikasit Bharat by 2047, and currently what food we are growing, how much by 2047, how much food has to be produced. There is a marginal increase in rice food grains, very less increase one time. But you can see that the horticulture in terms of nutrition, fruits and vegetables, 3 times more requirement. At present around 330, India needs 1,000 million tons of vegetables and fruits. Then rice, very limited 150 to 223, wheat 180 to 185. Then we want more millet crops, nutrient-rich crops. We want more milk, double, 3 times more fish and eggs substantially. For that, I think these are the technologies where Indian agriculture scientists in terms of mechanization and the water, which I will explain. And agriculture is also related to a lot of such SDG goals, which we have committed. But the beauty of the India is that around 300 crops are grown in India. We have almost world representative soil types. And world soil climates also we have, but we have world problems also in India. So how to see that the problem as an opportunity? I think a science and technology is a critical law, and agriculture is a state subject, and location specific technologies have to be generated. So how to prepare for Vikasit Bharat 2047 is the science policy, science and technology policy and program integration. What is the philosophy if the current production, increase in production has to be happened? What is the Swarnandhra's philosophy of sustainable agriculture? And in future, agriculture and sustainability depends on how we handle agri inputs like fertilizers, water, pesticides. How we handle natural resources like soil, water, climate, biodiversity, and climate, and policy support and ecosystem sustainability. First, most important threat to Indian agriculture is the climate change. See, even in Andhra Pradesh, when I was childhood, June 4th or 5th is the onset of monsoon. So, in the recent months, in fact, no rainfall in June or July, no rainfall, sometimes 1.5 months. We know that as a biologist or some of you may be knowing, in fact, 1 week delay in these things, how much production losses happen. And unfortunately, in India, rainfall has not declined for the last 70 years. What happened? Number of rain events have declined. Whenever they change, every week there used to be rain. Now in a 4 months rainy season, only 3 or 4 times rain. Whenever rain comes, 200 mm, 150 mm rainfall. How to protect the, in fact, food systems as well as agriculture and the crop in the field, how to be protected is important challenge. But it is happening everywhere. All the countries are facing more climate change. But India is the most vulnerable because we are the largest population. And we are the smallest smallholder farmers because technology implementation is very, very difficult when farm size is very, very small. And our sociology we also will not allow to spend more money on technology implementation. What is the global loss of climate change? You can see India, $1 trillion, the economic loss of climate change. So, India has also committed Paris Agreement and in net zero emissions. And Prime Minister declared that by 2007, India is a net zero. Agriculture currently is at 14 to 15% of GHG emitter. And the land use system, agriculture together is at 24%. So, agriculture has to contribute in the net zero emission targets. If not now, maybe tomorrow or day after tomorrow. And India is a very, very proactive in global climate change negotiator. Out of 192, 93 countries, India is one of the strong leaders. We need to prepare for climate change adaptation and benefits of mitigation. And recently, India hosted G20 countries. Of course, G20 countries have to play a bigger role because G20 countries collectively accounting 85% of GDP. All wealth is in these G20 countries. And 8%, 80% of world trade is that wherever trade and industry are there, they are emitting more GHG emissions. And two-thirds world population and emitting 85% of global greenhouse gases by these 20 countries. Therefore, we need policy corrective R&D and strong support. These are the two pictures which we made. This thing every year, Honorable Prime Minister visits IRI campus to interact, to draw the attention to that either protected cultivation or integrated farming system or millets. So these are the October, some of the photographs of October. And these are the net zero emission. Recently, we have developed a strategy paper how agriculture can contribute to net zero emission. But we can see that other country heads, some of the country heads don't believe. If Singh says that I don't believe there is nothing climate change happening, how to convince these people? I think some God has to come overnight, something happened. I think Mohan Rao sir told that some brain and these things, some change. I think only those aspects can change that gentleman so that climate change is really happening. I think need to prepare well. Otherwise, future humanity is affected. Second problem is that on climate change, the soils which were celebrating 75 years were not same soils. Even the Kakinada, Godavari district soils are not same. The carbon content is declining. Fertilizer application, the average fertilizer when we got independence is just 5 kilos per hectare. Now, the average Indian fertilizer application is around touching 160 kilos. Some of the states like Telangana, Andhra Pradesh, Haryana, in fact, 200, 300, 250 cases. Where it is going, utilization efficiency is only 30%. Remaining, they are entering greenhouse gases, nitrous oxide emissions, or groundwater pollution and the nitrate pollution. So you can see that 1950s, we have only nitrogen efficiency. Just like all of us, we need 16 elements. Plant also requires 16 elements. But you can see that the soil, or in 2025, all the nutrients are becoming deficient. And fertilizer imports are increasing. We are spending huge fertilizer imports. And one of the other important is that we are spending 2 lakh fertilizer subsidy. And mostly it is for urea. But when we have soil health card scheme and the sustainable agriculture, you can see that this is the blanket soil test based fertilizer application in the farmers practice, and 15% of the fertilizers are saved in that. People talk about organic farming. Of course, consumers preferring organic farming, but how to produce, where to produce is an important challenge. Sri Lanka, the story of Sri Lanka, which has seen that completely they banned chemical fertilizers. You have seen the consequences. I think there was serious food crisis. India, the population, we need to go slow and stepwise policy towards that bringing. Even my view that our chemical, I think best way is to integrate, not completely deviating the native strategies. And we have another concern where MS Swaminathan's pesticide load. I'm sure that all of you are aware that fertilizer. Tomatoes, one side sprays are happening, other side tomatoes are harvested, happily coming to supermarket. By evening we are purchasing, night we are making curry something and eating. And some of the fruits like grapes and other crops, be cautious. Especially leafy vegetables, Madam may be telling some more points. Leafy vegetables, green chili, I think let us be cautious how to treat with water and other things before consuming. So, but India, at Indian level, I think there's a conscious how to reduce pesticide load is important, how to replace these pesticides with natural products of customer. But if we don't use it, we are losing around 5 to 6 lakhs worth of agricultural products. We have to use it. But if we use there is a chemical loading into it. So how to balance these things? Then next important is the water. Dear colleagues, I have few more slides. And in India, food may not be a problem, but water will be the crisis. Already we purchasing 1 liter of water with a 20 rupees. In some villages, the 1 liter of milk is 45-50 rupees. So almost 40 to 50% we are spending for 1 liter of drinking water. So water will be the crisis. I think water resource is a very, very precious resource, and safe drinking water, quality drinking water. Agriculture is using almost 70% of water, available water. So important, already 54% of India and under water stress. Professor M.S. Swaminathan said that the field which is having one farm pond, the field which has that one vermicompost unit, that farm is a sustainable. Why M.S. Swaminathan said that sustainable agriculture? Because when climate change is happening, intensive rains are happening. The Swaminathan's philosophy that whenever intensive rains are happening, he collect water in these farm ponds. Whenever there is a mid-season and droughts are happening, he give micro irrigation protection. Dear colleagues, and I have seen in Anantapur district, a very dry district of Andhra Pradesh, one critical irrigation has given 40% more yield in terms of groundnut. So that is the secret of the success of one critical irrigation. So when that is very, you can see here, the Punjab, I think we have to take lessons of Punjab where rain revolution happened. In 1985, the entire Punjab is a blue color, groundwater is available. In 2017, you can see that the blue was replaced with the red, limited groundwater. The model prediction is that by 2037-40, in fact, Punjab has lowest groundwater. When we are going groundwater deeper and deeper, the water is touching buried material, then heavy metals are added and bringing to the farmland, then it is becoming cancerous. And you all know that the 2 districts of Punjab are declared the cancer districts. So let us see that in future, I think how it can be. Once it is harvested, we need to go for micro irrigation, not flood irrigation. Another important challenge in India is we are losing 30% food wastage. It is happening everywhere. Every plate you can see that any marriage functions, anything, but 60% of the food wastage is happening at a household level. Everybody has to take, otherwise, if this loss is not happening, how much agricultural land can be saved, how fertilizer can be saved, how much it can be, water can be saved. I think we have to think and strategize how the resources can be taken. Government of India, this is also not only economic loss, lot of methane emissions, 8% of greenhouse gases are coming from the food loss. It's not that one, dear colleagues, only rich countries are losing. Even you can see that South Africa 47, how much food wastage is happening. Everywhere food wastage is happening. I think it's a global problem. So one of the important is that lot of agri startups are coming, and you can see that the valuation processing is happening in India, lot of incubation centers. Another important aspect is that Madam must be telling how much oil is supposed to be used by India and how much human being. Broadly, they say that 1 liter per person per month, but we are in a year, it means that 11 to 12 liters. So we are drinking 18 liters, so that we are spending 1.8 lakh crores in oil imports to the India. Imagine that also costing healthcare systems, and this is the one of the climate resilient genome edited rice variety. Finally, it should be agriculture should be a root activity in India. Single farmer cannot handle the agriculture because of middlemen and market challenges. So farmer producing organizations are very, very critical here. So overall, the synergy agriculture needs corporates, policy, civil society. We need to work PPP, public-private partnership, for that strong agri education, strong R&D research and development is a back pillars are a critical need. So let me end with the laurels, accolades of Professor M.S. Swaminathan, as the Honorable Chairman said that he was a Padma Shri awardee, Padma Bhushan awardee 1972, Padma Vibhushan and a Bharat Ratna and first World Food Prize winner and so many awards. Then relating to last slide that how to see that Swaminathan's philosophy towards Vikasit Andhra Pradesh. Agriculture Andhra Pradesh is a very unique state. The length of Andhra Pradesh is around 1,000 kilometers, 942 kilometers. Big opportunity. All Indian climates are there in Andhra. If Vijayanagaram is different from Madanpalli and Anantapur. Anantapur is a very dry arid system, whereas Godavari has better water resources. And we have many crops. I think the legacy of the IRS, the MS Swaminathan can be easily taken to the Andhra Pradesh. Then if that is to be taken, Vikasit Andhra Pradesh, this is the last slide. The farmer income should be made as a business orientation because we need to bring that lot of commercial crops, industrial crops, and we need to go for the valuation. We need to go for farmer collectives and lot of agri-startup opportunity. Lot of resilient agriculture because we have a lot of coastline, frequent cyclones and floods are happening. Food security to safe food to nutrition, health, chemical agriculture to the soil health care and integrated nutrient management. Our agri education needs holistic and skill based education. And currently only governments are doing extension. We need to take to scale up the private industry as a co-partners. Cereal centric food system in Godavari district also a lot of rice, but there is a plenty of opportunity for commercial agriculture. Then only I think farmers' income can happen. Otherwise, rice is a safe crop, no risk crop, but less income. If you want to move high income and how lot of wealth creation through agriculture means we need to move from at least some portion of rice to the commercial crop. Marginal at present marginal cold storage. We need to intensive solar based storage with mechanization, which are critically required for Andhra Pradesh. And thank you very much.
You can interact or learn something more. Any two. It was a very interesting session, and after this, you will have the last and final, but a very interesting session by a very interesting personality. Don't miss that. In this particular session, 4 persons were referred mainly in agriculture. One is Dr. Norman Borlaug, who is father of Green Revolution. He got Nobel Prize. Second one is M.S. Swaminathan, Bharat Ratna, which is like Nobel Prize only for us. Another person is N.G. Ranga. Another 4th person is, I was mentioning M.V. Rao Garu, who belongs to this area. He got Padma Shri for his work on wheat. He gave a beautiful presentation on agriculture, wheat production in the same area where Sir was presenting. All these 4 persons, do you know the commonality of all these 4 persons? All these 4 persons, they lived for 90 years plus 98, 96, 95 like that. So everybody present in this hall who is going to participate in the last session of the day, lecture of this day also, will live for minimum 95 years. Any one person? And he is a very lucky presenter that his presentation was listened by another Padma Shri awardee, Dr. Chandrasekhar Garu. We are all very lucky and blessed that he has come and participated in this session.
Thank you.
Otherwise, maybe at the end of both the sessions, we can ask one of them. May I now invite somebody is introducing me. Dr. Sridevi Annapurna Singh. May I invite you, madam? Morning, you stole our hearts. Okay. Okay. So, we'll be very happy if madam is present for felicitating our speaker, Sri Dr. C.H. Srinivasarao. I invite you to please come by the comment. Krishna Prasad is there. Your Atma is there. May I request you, sir, kindly can you come to, please, Dr. Chandrasekhar. Okay.
Yes, yes, sir, please. Sir, sir. Yeah, please. Thank you.
This is Padma Award, Conclave Sanjay Gandhi, UBC member, and Telangana. I hope everyone knows about the Conclave Foundation. Great man. I request him to say a few words, address the guest. Afterwards, Madam Sridevi Annapurna session will start. Madam, please, please, no, please, please. I just came here by the honestly. Please, but very nice presentation. And there are real times we are going to face in the future. So the whole world is getting ready or should get ready, but unfortunately not. So we all should think what we can do as a single individual and then as a community, as a group of citizens. We should all try to think and plan something to save the planet for the future generation. I strongly feel about it, especially after doing stop here. I know it's all happening, but he gave very nice, vivid presentations and what to look at the future and how things can be improved by our own efforts. No single individual can do this miracles, but collectively, everyone should participate in this and make it. Thank you for giving me this opportunity.
Before I invite Madam for her talk on Indian Thali, ancient wisdom and modern diet, can India be the sustainable, healthy and wellness football for the world? Very, very interesting topics. Before she starts, let me quote 3 sentences. One is Muhammad Yunus, Nobel Prize winner, said, any problem, any business, including research, is not for money. It is for solving problems. So today, one big challenge is always food scarcity, food, health, etc. is a very big challenge. Top 10 challenges in the world. So this topic will address to that particular problem, and research is to solve a problem. The second point is domain knowledge always expands, enhances, enriches, and advances at the intersection of disciplines. So your knowledge in your own subject also, whatever topic you are doing research, it enhances, enriches, advances, and expands. Only when you show interest in other branches also. And the third point is, Karl Marx said, diversity is strength, and strength of India is her diversity. So only when our knowledge is diversified, then only we can become a very strong person. And to enjoy the knowledge of a different field, you should give your mind a lot of freedom. That is why in Latin there is a very beautiful statement that says, fortis sette libere. Forte means strength. Libere means freedom. Only when you are strong, you will have the freedom to do and express. Otherwise also correct. Only when you have freedom to do and freedom to express, then only you will become strong. So whenever a new topic is coming, instead of saying that this is not connected to my research, immediately if you can understand and appreciate the depth of that knowledge and its application to your own domain knowledge, then definitely it helps me a lot. So let us welcome Madam for her talk.
Madam mic చూడు. Why Janathan mic చూడు madam కి.
Dr. Sridevi Annapurna Singh Garu. Dr. Sridevi Annapurna Singh is well-known food scientist with more than 34 years of experience in research and leadership at Center for Technological Research Institute, Mysore. She completed her MSc in food technology in 1988 and PhD in the food science from the University of Mysore. After working in industry, she joined CFTRN in 1991. Over the years, she became the chief scientist and head of the protein chemistry and technological department. Later, she served as the director from 2021 to 2025. Dr. Sridevi Garu has published more than 65 research papers, reviews, and book chapters. She holds 12 patents, including a patent on UX, and has guided over 48 PhD and PhD students. She had very important leadership positions at national level and currently serves as a service scientific and food safety company. She is a fellow of AFSC and received many prestigious awards and honors. Dr. Sridevi Garu continues to contribute actively to the food science research and policy. We are truly honored to have such experts among us today. Please join me warmly welcome Dr. Sridevi Annapurna Singh Garu. Once again, very good afternoon, and I'm sorry, it seems two lines are getting cut in my presentation, so along with you, I will also not see the top line and the bottom one, so forgive me for that. I thought that I will, all of us love food, and all of us cannot live without food. So it is a very, very important basic need for us, but I would try to really connect what is that we have learned over the years and what is the legacy we have got from our ancient people, what we have inherited, and actually we do not connect diet and disease many times. It was COVID which brought it to the fore, and we know now that diet and disease or diet and health are very much interlinked. And I'll just give you a few examples. But in India, in spite of, the last four lectures were so wonderful, and especially the previous one really brought out how agriculture has improved in the country, and we are among the top 5 in agricultural commodities in the world. But it is not just enough to produce the food. We need to value add because food is perishable. And unless we do that, the farmer will not be able to get a good price for the crop. We have to create the demand through value addition. And that can only happen if there is post-harvest processing of the food, because like I said, it is perishable. So again, the previous speaker has touched upon this, but why is this such an important thing for Vikasit Bharat? Like we have heard the previous speakers, India cannot develop in only one area. We need to cover everything, only then overall development will be there. Otherwise, the weakest link will be our problem and we will not be a developed country. So since I have worked in the area of food, especially, I will only concentrate on that. And if you look at it, it is one of the most important activities in many of the countries, because 25% or like in India, more than 40% of us are dependent for our livelihood with producing food. And around 16 to 19% of India's GDP comes from food production, that is agriculture. So, but on the flip side, if you see, we need a lot of land. And because of the wastage or because of non-production, we are wasting a lot of food, and that is leading to a lot of greenhouse gases. Plus, we are also losing biodiversity because many of the forests are getting cleared so that we can use it for agricultural purposes. On the other hand, many of us are eating the wrong kind of food. There are at least 200 million in India alone who go to bed without a meal. They are going hungry. On the other hand, many of us are eating the wrong kinds of food. Your cells want vitamin A, you eat carbohydrates. Your cells are asking for omega fats, you are using something else. You need proteins, you are eating something else, which the cells do not want. So perpetually we are hungry. That is, our body is craving for certain nutrients, but we are giving it something else which it does not require. And just like if if you have 100 rupees in your, you're given 100 rupees, you spend only 50 at the end of the day. You don't throw away the other 50 rupees, right? You would either put it in the bank or you would save it. Our body does exactly that with the extra calories that we are eating. It stores it. And where does it store? Right around our abdomen in the form of fat, which means that all our organs are covered with fat, which is a very active fat. It is hormonal in nature, and therefore it is creating havoc and leading to these so-called non-communicable diseases or NCDs. So we need to rethink the way we are eating. And as the previous speaker did say that nutrition and the soil nutrition is also decreasing. So we need to ensure nutritional security of our population. But at the same time, we have to reduce the amount of natural resources that we are using, be it land or be it water. All of this is becoming more and more precious, especially in India. So it seems there is a conflict between the version I have worked with and this. So some of the slides may look a little scrambled, but over the years, what has happened? Why was it that our grandfathers and great grandparents were very healthy? We keep hearing this in our family that even at the age of 80, they were able to work on the farm, they were able to do a lot of physical activity, whereas at the age of 40 or 50, we are just dropping dead or we are getting some kind of a chronic disease. So what has happened is food processing, when you look at it, we were eating the natural grains in the beginning. If you were in the Godavari belt and you're growing a lot of rice, it was hand pounded and you were cooking it and you were finishing it off by the end of the day. So what is there in that grain which is not present today? All the bran layers were there in that grain. These bran layers have a lot of vitamins, minerals, apart from the macronutrients, which is either starch or protein or fat. There is very little fat in many of the cereals, but then protein and carbohydrates are also there. Now, why should we remove it? When modern times, what we have done is we have started polishing the grain. The reason is that there are certain composition in the bran which does not allow the grain to be stored for more than 15 days or 20 days. But you want the grain to last for at least 6 months, maybe 1 year. So what happened? The processor started removing these layers which reduce the shelf life, what we call as shelf life. And therefore, we started eating only carbohydrate, mainly starch, and very little protein which is there in that grain. And therefore, we started losing out on many of the nutrients that we should have been eating. And today, whatever you are eating mainly lacks most of the micronutrients that your body requires. Apart from that, of course, we are sleeping less. We have a lot more stress. We are all the time worried. We have lack of physical exercise. Since morning, we have been only sitting in the seat and we have the air conditioning going on. So we are not really burning the calories that we have eaten also, which means that our body is going to store all the extra calories that we have eaten in the very delicious lunch that will provide as fat around our organs in the abdomen. So also processed food, what has happened is that because they have to look like the food that is made fresh at home, we have added a lot of salt, fat and sugar as well as used food additives to make it mimic the food that is cooked at home. So over a period of time, it is not just that if you eat it once, something will happen. But over a period of time, what happens is slowly your body starts reacting to the excess. Because if they have to be eliminated, there are two organs in your body, the liver and the kidney. They have to remove these toxins from your body. So when you keep overloading, finally there comes a time when it cannot be expelled, and that is when you start seeing the effect of the kind of diet that you have had. But on the other hand, it's very convenient to have processed foods. It is cost effective and it is very, very tasty. I'm sure even after I have spoken, if I keep a pizza and Coca-Cola over here and keep hand pounded brown rice and rasam over there, many of us will go towards the pizza because we often want taste. But unfortunately, this is what happens. You can see how the structure of the body changes and slowly abdominal fat keeps increasing, which is a very dangerous kind of fat because it prevents your pancreas from working properly and releasing insulin and so on. So even in India, you see that we have become the diabetes capital. We are number one in cardiovascular disease globally. A lot of deaths in India are not because of accidents, not because of murders, but because of the kind of food that we are eating, which has led to a disease and the complications of that disease have led to death. But health has become very, very expensive. Nobody wants to go to the hospitals, right? So it is important for us to know how you can use this diet to see that you remain healthy, which is what I want to communicate to you today as to how India can use its ancient wisdom and how our people were able to survive for a very long time with good quality of life, and how this can become a global solution for the rest of the world also. So today we have realized that even in a family of 4, it is not necessary that the same diet is good for all 4 of us. We need to actually, we are moving towards personalized nutrition. Somebody is diabetic in the family, they need a certain diet. Somebody has a metabolic disorder, they need a certain diet. Maybe somebody is pregnant or lactating, they need a different kind of a diet. So based on this, today we call that personalized nutrition, based on the genetics, based on the environment, based on the health status, based on the activity that you are having throughout the day. All of these factors have to be taken into consideration to actually decide what is it you have to eat. So since it is a very vast area, I'm only going to stick to these 3, that is calories and proteins. And also, please remember, like I told in the beginning, the environment is suffering because of agricultural activities. Cost is important, convenience is important, nutrition is important, health is important. So considering all of this, I'm only touching upon these 3 aspects, that is calories and proteins, functional foods, as well as waste to wealth. So this has already been covered by the previous speaker. That is, if you look at the amount of grains or milk or eggs, all of this that we are producing, we stand among the top 3 producers in the world for most of the commodities. But since we are not processing most of the $60 billion that was told as the export of India to other countries are all after primary processing. What do I mean by primary processing? You are growing rice, you bring the rice, the paddy, clean and millet and export it. But it goes to maybe China or some other place. They convert it into a product and sell it back to us at 10 times the rate. So although we are producing it in our country, we have become a raw material supplier rather than value addition to the crop. And this, if you look at the world map, even Thailand, Vietnam, Malaysia, Indonesia, doesn't matter how small the country is, they are all value adding to it and they have a large amount of processing done. Whereas India, in some sectors, only 2% is processed. Only in dairy, you will find that 35% of the milk that is produced is processed. So there is a huge scope for all the youngsters sitting over here to work in the area of food and also to get into processing, because although the farmer has sweated so much of fertilizer has gone in, we wait for the rains and all of that. But because we have not processed, the food has gone waste. So some of the opportunities for India, like locally also, I know in my childhood I've eaten a lot of very indigenous, unique crops that are only present in this part of the country. There are many such that are there which can have unique nutritional properties. They may give very unique health benefits, but we need to tap on that. Unfortunately, because the farm holdings are very, very small, what happens is we have maybe 2000 varieties of rice, but we can only supply 50 kgs. Now, which factory can run on 50 kgs total? Per day, you need 5 tons, 10 tons, maybe 50 tons. We don't have that raw material. Now, if you mix all the crops, what happens? Something will have higher amount of husk, something will have lower amount of husk, some may have the nutrients in the bran layer, some may have it in the endosperm, and so when you are milling it, it is lost. So there is a lot of overheads in the factory because of that. I'll tell you, if you are liking a Pepsi chip, the chips, the potato chip, including the soil, what is the condition of the soil, how much of water is required, what is the quality of the seed, when it should be harvested, what is the thickness of the chip, how long, at what temperature it has to be fried, which kind of oil has to be used for that. Everything is controlled by the factory. Everything is controlled by the Pepsi company. Here, our factories have to take what is available in the market. So how do you compete globally if you don't have control over the raw material? And secondly, we are using more fertilizer, more pesticides. Most of the countries have specific standards. What should be the residues in the product? If the processor does not have control over how much of it is there in the raw material, then it gets rejected finally when it is imported into another country because the laws are so stringent. So, one lead that we have is millets. We used to eat a lot of millets earlier, but today, because thanks to the green revolution and the various techniques and machinery that is available for milling rice and wheat, but not available for millets, we are not consuming that kind of what should be consumed. 330 million tons is only rice and wheat majorly. Millets, we have around 17 to 18 million tons per year. How do you feed a population of 1.5 billion? Forget about the 8+ billion people who are there on earth. But it is a healthy crop. Second, it does not require too much of water. It is a drought resistant crop. So it is only very minimally polished and converted into a product. So it has all the health benefits that are missing in polished rice. So even if we can substitute 30% of our meal with millets, you would be healthier. But we need to redo the calculation of how to process. And that is where the opportunity lies because India is the largest producer of millets in the world. And we have so many regional recipes which need to be scaled up, which need to be automated, and then you have a winner because a lot of people want to stay healthy through diet. So these are some of the challenges that you see in millet promotion. You don't have dedicated machinery. We use rice machinery, we use wheat machinery, which was developed abroad, and we try to fit in the millet, which is a different size, a different kind of, it is circular, it is small seeds. They are not like rice or wheat, which are oblong. And again, millets have a very limited shelf life. If you think of jowar, hardly after you made jowar flour, you'll have 15 days. After that, it goes bad. So we need to understand how to control the enzyme activity, which will then prevent the jowar flour from going bad. Another thing I would like to say is that although we have been very, very successful in increasing our agricultural produce, we have not looked at, there has not been a marriage between agriculture and food technology. That is the processor. So what has happened is although it is wonderful to cook the rice, it may not be the best rice to actually mill. There may be a lot of breakage.
That also needs to be looked at. So now is the time when both have to get married and optimize the way the product is produced. We need a lot of convenience products, and then we need scientific study, because there is a lot of epidemiological data, and there are a lot of grandmother tales also as to the health benefits of many of these grains. Many of us eat it at a certain time and so on, but there is no scientific validation of that. And unless we do that, the world will not listen to us. I may listen to my grandmother, but it is not necessary that the world will listen to my grandmother. So scientific validation is very, very important, including clinical trials. And then we have to, like I said, there is low production, there are varietal differences. Recently, when I was in CFTRI, we had looked at the nutritional profile of 130 varieties of different millets. And we find that even the iron content varies such a lot. So we need to really classify which variety to grow if I'm giving it for a person who is suffering from anemia. If I'm looking for a child nutrition, I should not give something which is having a lot of anti-nutrients. So if I'm looking at geriatric nutrition, then I have to take a different variety. It is time that we do these exercises. Now, why should India even look at the food space to really make a difference? Because any market you look at, whether it is infant nutrition, sports nutrition, or enteric nutrition, everything runs into billions of US dollars. Unfortunately, we are only at 2% of the world market. So we really need to understand, and we need to work on these gaps and challenges to turn them into opportunities so that we can become leaders, because we are the top producers of most of these raw materials that are there. Now, coming to protein, I have worked in the area of proteins for a long time. The world is looking at plant proteins. All of you are, some of you may be vegan, some of you want only plant proteins, not animal proteins, for different reasons. But environmentally also, to farm animals, you need a lot more water. A lot of the forest has to be cleared, and a lot more greenhouse gas emission is there in animal farming. So the world is looking, because the supply chain has improved so much, even the cold countries are looking towards plant proteins. So again, here if you see, the cost economics is much better when you look at plant proteins as compared to animal proteins. Why is protein so important? Any function in your body requires protein. Today I learned that even for robotics and AI and so on, Dr. Mohan Rao explained that protein can be used as an energy source. So it's wonderful what proteins can do. And it's only the 20 amino acids that are reassembled in a different conformation to get proteins which have different functions. Some proteins are enzymes, some are associated with DNA, some are, transporting your nutrients to the cells, and so on. You have all these varied. So what exactly is it that makes a protein have this versatility? It is the amino acid composition. You have these 9 essential amino acids. What are they? These are the amino acids which are the bricks of that particular protein, which your body cannot make. They have to come from your diet. So if your protein is deficient in these amino acids, then that protein is not utilized as protein, but can be converted to fat, and you are not eating sufficient protein. And because the protein has so many functions, ICMR has said that for an adult, which all of you are, at least 0.8 grams per kg body weight has to be taken every day. So if you weigh 60 kilos, 48 grams of protein has to be eaten, which in a vegetarian diet is a little difficult. Plus, it has to be balanced and have all of these as per the requirement of the body. And we have all these sources that are readily available, which probably no other country can claim that they have sufficient quantities of plant protein sources, including seaweeds and algae. But here is the catch. Though we don't want to eat animal proteins, the animal has already eaten the plant seed, it has removed the anti-nutrients, and it has reassembled the amino acids which are essential in a certain conformation, which is very near to what a human being requires. So when you eat an animal protein, it is very easily digestible. When you look at a plant seed, which is also having higher protein of maybe 23%, soya has around 40%. There, the seed is made for a new plant to grow. So the mother plant has not made it as per our requirement, but as per its child's requirement. Second, to defend that seed from not being eaten by insects or by fungi or by humans or by animals, a lot of defense molecules are also over there which hinder the digestion so that you will not touch that seed. Also, when the seedling is growing, maybe it requires certain specific nutrients in larger quantities. Then these so-called anti-nutrients are broken down as a source for that plant till it puts its roots and shoots and starts making its own food. So plant proteins have lower digestibility and they have lower absorption. They have to be cooked in a certain way for a certain time to make sure that the digestibility can be improved. And in the food matrix, we need to combine many foods to see that we get all the essential amino acids as per our requirement. But plant proteins are good because they have higher fiber content. Many of us had stopped eating fiber. We were polishing our grains and eating and thinking it is only nutrition. But today we know fiber is equally important because our gut microbiota's food is fiber. And for every human cell in your body, there are 10 microorganisms in your body, which means it is the microorganisms which are driving you towards health or disease. So you have to take care of the good bacteria in your gut. So one way of removing these is through the preparation of protein isolates and concentrates. And you can use processing techniques, like I said, like fermentation, germination, pressure cooking, and so on, to inactivate these so-called anti-nutrients and increase the nutritional availability of many of these beans and other seeds that have the higher protein content. There are many applications for using these protein concentrates. We are all familiar with whey proteins. Many of the sports persons are mad after that. They consume a lot of protein because of that. Then comes the substitution of meat. So many people who are addicted to animal foods want that particular texture and flavor that comes with, which is associated with that particular food. So we need to texturize these plant proteins so that they are resembling the animal protein that they are eating, and people can then shift towards plant proteins. Then enzyme modification is another example where you use external enzymes to break down the protein. Most of plant proteins are very high in molecular weight. They are very tightly folded, which means that our gut enzymes are not able to actually access and break them down. So you use external enzymes to break it down and then prepare the food, which means that they become much more amenable, maybe for health purposes. You know, earlier we used to see in the old black and white movies, whenever a couple gets married, the girl comes in after the marriage into the room at night with a glass of milk. Today, we know that milk gets digested, a lot of peptides are released which relax a person and have a peaceful sleep. Science has proven that. So the ancient knowledge is drink a glass of milk before you sleep. But science today has told that this is why you have to do it. So there are some health benefits of certain proteins. And enzymes can do it also for the elderly. Geriatric foods can be prepared because many of them have lesser amount of acid in their stomach. They cannot digest it. Sometimes they're taking a lot more medicines, therefore they do not want to take certain foods. And their sensory organs also, especially the tongue, it does not get stimulated and want to have certain foods. Sometimes they lost their partner and are dejected. So for all of them, these kind of foods, sometimes they don't want to go near the stove. So if you have foods which specially cater to them, that will be a big bonus so that their quality of life is not affected. So again, what I am saying is what was 5,000 years ago recorded, that food is what makes you as you are today, and our body is a product of the food. So the Indian traditional food system was very varied. We had this huge plate with a lot of things on it, which ensured that we had menu diversity. We also had certain beliefs that if it is Amavasya, you don't eat this, you eat another food. If it is some other thing, you should not eat. For a death ceremony, you don't eat certain foods. All of this ensured that we ate different kinds of food so that what is lacking in one is made up for by the other and your body remains healthy. Unfortunately, today we make one dish in the morning, have it for breakfast, pack the same in the dabba, come home and eat the leftovers in the night. So although your body requires a lot of nutrition from different sources, you are not giving it. So it's time we have a relook at our thali to really understand what is it we should be eating, at what time we should be eating, and how we should eat it, how it should be prepared. So there is a lot of knowledge that is available, and many of we've stopped listening to our mothers and grandmothers, right? Because we want to live on our own. We like Swiggy and we like to go to the malls, but they do have a lot of knowledge which needs to be captured and scientifically proven. So these molecules which are there in the foods, they are called nutraceuticals. That is, they're coming from nutrition, but they have a pharmaceutical kind of application. So they are nutraceuticals. And many of these Ayurvedic products do have these molecules in ample amounts. And there are a lot of fermented foods also in Ayurveda which have this effect. But we need to relook at them and scientifically validate whatever it is. So again, there is a huge market for this. It runs into billions of dollars. If we don't do it, America will do it and they'll capture the market, they will patent it and we will lose the knowledge. So there is a huge market potential. Again, the food that have the nutraceutical is called a functional food. And combining this by eating such foods and becoming well beyond health is called wellness. And that is what we are aiming today. That we don't want just health, we want wellness. You wake up happy, you want to do things, you are cheerful. All of that is under wellness. So if you see, although we have this wealth of knowledge, India comes very low in the ladder of capturing markets. There are many ways in which these nutraceuticals protect us, and we have many foods that have been identified, including spices. However, there are many gaps also. We need to standardize the variety. We need to do innovative processing so that the bioactive molecule is not broken down and remains in the food. And we need to even have traceability from which part of the country it has come. And we need to do global branding, that it is a product of India, and this is what its effect will be if you take it for 2 weeks or 2 months or 2 years. And all of this needs to be developed, including the supply chain, the way it is being stored and so on. I'll just give you a small example of what my student did. The sesame oil is what you use for pickles. It is believed in Andhra that sesame oil has long shelf life, so the pickles even after 2 years won't spoil. Now, there are these three molecules that are there in sesame seed. One is sesamin and the other one is sesamoles. When you heat the sesame oil, it gets one of these molecules, that is sesamolin, gets converted to sesamol. And this is a very high antioxidant activity it has. And in our body, every day, because of stress and because of pollution, because of what we are eating, we have a lot of inflammation, which is there existing in our body, which causes diabetes, which causes cardiovascular disease, and so on. For a small amount, it is okay because our body requires it. But when there is an overdose of these radicals, it leads to inflammation. So what is happening there? In the cell membrane, the arachidonic acid is released by phospholipase, and then it enters enzymatic cycle, either lipoxygenase or cyclooxygenase, that finally results in inflammation. So if it is less, it is good. If it is too much, then you have a pathological condition. So when we use this molecule, sesamol, which is there in sesame oil, what we found is that this particular enzyme that will convert and release these free radicals and lead to inflammation, that is suppressed. So lipoxygenase is a molecule wherein it has a cofactor called iron, and that is in its 2+ form, which is reduced. In the presence of small amount of hydroperoxide, it gets converted to an active lipoxygenase where the iron gets converted to the 3+ form. This is what will make it very active and cause inflammation. So what sesamol is doing is removing this hydroperoxide, and therefore the reaction cannot go forward. So this we are able to prove. Like this, if we are able to prove for other observations also, we have very good set of foods that can be marketed properly globally. So we need all of this. Again, I think it was Dr. Ahmed who told about what all needs to be done, so I will not talk about it. Now coming to the, if you see food waste, it is very, very huge. I'm only talking about one underutilized grain, which is called Niger seed, and it is hardly utilized for edible purposes. Mainly, it goes for lamp oil because the hull is not removed. There is 17% hull in it. And this oil is used as lamp oil because it is dark and viscous. Further, it is only grown by small and marginal farmers who do not have too much money and are rain-fed regions and require the rains to come properly for their crop to grow. However, it is a very good quality oil, and the cake that is there only goes for animal feed, even though there is 50% protein in this, because the hull is not removed, it is dark and not good to taste. So where is this used? It is exported to Singapore or other European countries to use as bird feed when our people do not have sufficient protein. Now we looked at that hull and we tried to remove the pigment that is there, which is black in color, and identified it as melanin. So a waste material which is dark in color has melanin that sells at US dollars of 455 per gram. If you take 1 kg of hulls, you will end up with 15 grams. So please do the math. You don't need the oil at all. You will recover your money just because of the pigment. So what we did was to recover this pigment by various methods and classify it. And then after identification, we co-extruded it to make a film. And because of the high antioxidant activity of melanin, we use that to package very high value agricultural produce like strawberries, which hardly stays for 2 days. But when it is packaged in this film, even after 5 days, it was barely good. So again, not only you are recovering a very high value molecule, you can also use it to save other foods. then of course, many of the agricultural waste can also go into all of these. But like I gave one example, there are many natural bioactives that can be actually isolated from food waste. Probiotics, again, all of these waste molecules do have a lot of carbohydrates which are not starch. And those are the food which are either insoluble fiber or soluble fiber for our gut microbiota, which is again a huge market running into billions of dollars. And they can be recovered for right now, India does not produce any fibers. We are only importing, whether it is Yakult or some other kind of a product, it is only being imported. So there is a huge market over here, and India can overtake the world for that. So I have already said most of it, but just to reiterate, we produce a lot. Unfortunately, we are doing the farmers, we are giving them a raw deal by not converting and not adding value to what their sweat and blood has produced. It is high time that food technology and agriculture go hand in hand to make sure that we value add and we increase our GDP, we provide a livelihood and encourage our farmers to grow more and more food rather than leave their profession and come into the cities. So if India has to be the global leader, then we need to look at health, nutrition, make sure the food is adequate, safe, healthy, and then look at the economics. Only when you process optimally, you can do all that. Look at the social and cultural acceptability, not only in our country, but globally, so that we have a good lead in that. And of course, environment and sustainability is very, very important. Thank you.
Thank you very much, madam. We will end this session, last session with 2 questions. Minimum 2 questions and maximum 2 questions.
Let me ask it. I mean, article, I think. This I have the classification of amino acids, right? Essential and non-essential. But in the presentation, I have this 3 types. Conditionally non-essential.
Yeah. So suppose there is a person who has a metabolic disorder like phenylketonuria. So phenylalanine is an essential amino acid, whereas tyrosine is not essential because our body can convert phenylalanine into tyrosine. But people who have PKU, they are unable to metabolize phenylalanine because of the absence of a certain enzyme. In which case, tyrosine becomes essential. So you have to minimize the phenylalanine in their body to their daily requirement only. You cannot give excess. And then you have to give tyrosine so that need is also met. So like that you have conditional requirements in certain conditions.
Another question, you have presented that pressure cooking may reduce
Nutrients, yes.
actually, but I have listened to a case where they are quoting Varpada's theory. Pressure cooking may consume nutrients. Instead of cooking pulses, it masticates. What do you say?
So usually there are many anti-nutrients that are present. Okay. One class is oligosaccharides. You have raffinose family of sugars, which our body cannot digest. Those are soluble and they will come into the soap water.
Talked about pulses, not
Pulses only, I'm telling. Then there is what we call as trypsin inhibitors. There are 2 kinds, Baumenberg and I forget the other one. Both of them are very, very stable. In that, Baumenberg is having 7 disulfide linkages, which has to disrupt. So when you look at a protein, I did not talk because of want of time. I'm sure I've exceeded in spite of it. But what happens is the protein amino acid sequence and then its particular fold will determine its activity. So if you want to disrupt the entire structure, only then that activity is lost. Now, a protein has 7 disulfide linkages. It is very rigid to digestion. So you have to break those bonds and misfold that protein for its activity to be lost. And that is what pressure cooking does. So if it is too dull, 2 whistles of the pressure cooker is enough. If you are cooking rajma, you go up to 6 whistles. If you go up to this chole, then it is some other thing. All this over a period of time, our mothers have already standardized. They don't know why they have done that. They only know that you will not get gas in the night. You will not be able to, it will not be very hard. All of these they have standardized, but actually what is happening is this. So often you soak the dal or the seeds and then at least 3-4 times throw out that water. So the oligosaccharides will go. Then you pressure cook it, you add a pinch of bicarbonate. Again, you can denature many of these anti-nutrients much faster. If you take an infant before all this Cerelac and all that came, ragi, you sprout it, remove the hull, remove the shootlets and the rootlets, finely grind it, through a muslin cloth it is sealed. Because the infant does not need that kind of fiber. They want
Can an adult take all those hulls and?
yes, you require to manage your weight, otherwise you will.
I'm not in a position to manage because
You are fine. I'm not
still
saying
this is
for
a
you.
request.
What I mean to say is in general.
This is not a request. I mean, this is not a question.
So you require at least 28 to 40 grams of fiber per day.
I'm taking it. చిన్న మాట మీకు ఏంటంటే తెలుగులో మాట్లాడాలంటే వినాలి
మాట్లాడండి.
తెలుగులో మాట్లాడాలంటే
మాట్లాడండి.
ఏదన్నా చెప్పండి తెలుగులో ఎందుకంటే మీరు తెలుగు ఆడుకోవచ్చు కాబట్టి తెలుగులో మాట్లాడితే వినాలి థ్యాంక్ యూ
ఏం చెప్పమంటారండి?
Thank you.
చాలా సంతోషంగా ఉంది కదా.
Last and luckiest, one more. రీసెర్చ్ లో అడగాలి, క్వశ్చన్స్ అడగాలి. సూర్యుడు ఉదయిస్తే సూర్యోదయం అంటారు, చంద్రుడు ఉదయిస్తే చంద్రోదయం అంటారు, ప్రశ్న ఉదయిస్తేనే జ్ఞానోదయం అంటారు. Ask questions and get enlightened. We should really appreciate Madam. Morning in the inaugural session, she was very active, highly dynamic and full of enthusiasm. And now, the last session, last few minutes of the last session also, she is with the same energy and enthusiasm. If there is no question from anybody, we will come. Thank you very much, madam. This program ended with a very beautiful, interesting topic, and that is food and agriculture. On a lighter side, for the nation, agriculture is very important. In personal and professional life also, agriculture is very important. What is this agriculture? Agriculture is the good culture of a good husband to agree to whatever wife says. And in profession also, a good employee is one who agrees to everything that his boss says. So if we can develop that agriculture, then life will be very beautiful, personal as well as professional.
The men only agree, they do not follow or do. But I'm sorry for the sites where it's not. I think there was some kind of a conflict.
No, your very presence from morning to evening, it is a very big highlight of this. And thank you very much. As a take-home point, let me just share one statement that says, today's wastage is tomorrow's shortage.
Yes.
If we waste any food today just because we have a plenty, then take it for granted, tomorrow we will suffer, or our next generation will suffer due to shortage of that food, whatever it is. So today's shortage, today's wastage is tomorrow's shortage. I profusely thank JNTUK KAKINADA University, Honorable Vice Chancellor sir, Krishna Prasad, other organizers, Srilat Mari, etc. for giving me this opportunity and thank you very much.
Thank you, sir. Thank you, madam.
Just wanted to say that the person who taught me is also sitting in the audience, which I'm very happy that Professor Ramakrishna is there, who taught us food engineering. And while he works in CSPRI, he has brought his technology to this campus also, I understand. And he has been instrumental in starting many courses. So food technology will flourish.
Right, thank you, friends. Now, yes, book releasing function is there. Ragananda Lahari. I think I have pronounced it correctly, sir. Thank you, sir. Friends, afterwards, after book releasing function, cultural programs are there. Afterwards, for the participants who participated in the technology conclave, dinner is provided at guest house. Okay, please.